Joining. This is a training course in the Work Package 1, Selective Breeding for Robust Fish of the EU-funded project PerformFISH. PerformFISH is an industry-driven project, and it is set to increase the competitiveness of the Mediterranean aquaculture sector by tackling biological, technical, and operational weaknesses that underlie the stagnation of the marine fish production in the last decade, while addressing social and environmental responsibility and contributing to EU sustainable blue economy and food security. So it's a consortium that includes 28 partners, of which 12 linked third parties that are distributed in 10 countries. These are the three different levels on which the consortium is working on. So we have studies that go from the broodstock to the hatcheries and on-growing, studying each time different aspects of the sea bass and sea bream production. Of course, the consumer and society perception is everywhere in the consortium, in the project, and the objective is to provide tangible results at the end that can be used both by the industry but also to make a profit for the consumers and the society. We are aiming at improving KPIs like growth rate, mortality, and feed efficiency through a series of studies that have to do with marker-assisted selection, epidemiology, vaccines, and this is different aspects that are included in the different work packages of the project, starting from the first one, ours more or less, the work package that has to do with the selective breeding for robust fish, going also to work package 2 for juvenile quality, for survival and growth potential, work package 3 that has to do with the health issues and the parasites and the viruses that are a problem to the Mediterranean aquaculture. The work package 4 has to do mostly to feeds and try to implement novel ingredients into the feed industry for bass and bream. And on the right side, we can see that in the PerformFISH project, we have on our side, the associations, the major associations of the Mediterranean countries like the SFMN in France, the APROMAR in Spain, API in Italy, in Croatia, and FGM in Greece. The general objectives of the work package 1, so selective breeding for robust fish, were first to develop a practical SNP genotyping tool for marker-assisted and genomic selection, to develop new phenotyping methods for important traits like feed efficiency, disease resistance, and fish shape. Third, to evaluate genetic and genomic variation for these traits and produce fish for genotypes by feed interactions. And last, produce case studies to include genomic evaluation in European sea bass and gillhead sea bream breeding programs. The specific outputs now for work package 1, and this is the more or less the last slide of the introduction, is as we said, to design and develop a SNP chip tool that was going to be validated in experiments of the PerformFISH project to analyze family and genomic variation for feed efficiency, estimate EBVs for vibriosis and parasites infections, study the genetic architecture for morphological abnormalities in gillhead sea bream, estimate genetic parameters for several growth and or morphometric traits, and compare these EBVs under the different models, and afterwards to estimate genetic parameters and low-density genome scan for SNPs associated with the VNN resistance in C-BRIM. And last, try to do a global genomic evaluation compared to that will lead to genome-wide association analysis using the SNP tool that the project has developed, but also with information coming from different projects like the MedAid, but also we have to acknowledge also the French Genesee project. Okay, these were the 12 partners that were working together in the last 4 or 5 years and more, if we also count the time since the project started to be designed. And we have partners from Greece, Italy, France, and Spain, and Croatia, together with the respective associations of fish industries from these countries. This is the program, the agenda of the today's training course. I'm going to follow what has been proposed. So we are aiming at 35-40 minutes presentations, and we're going to accept and try to reply to few questions that are going to be proposed by the attendees. And after each session, the morning and the afternoon one, we're going to have somewhat bigger question and answer session, trying to wrap up and be okay with all or more of the questions that are going to be asked by the audience. So I think that I have respected well the time, and I'm going to start with my presentation with the title of Genetic and Modern Genomic Approaches in Aquaculture. So I'm representing the Institute of Marine Biology, Biotechnology and Aquaculture, that it's part of the Hellenic Center for Marine Research, and we are based on the island of Crete in southern Greece. I'm going to show you a few slides of the current status of the aquaculture production and why the genetic markers and the genomic technologies are needed. So we all have to bear in mind that the aquaculture is the fastest growing livestock agriculture production sector, with an average annual growth of approximately 9% till the 70s, as compared to almost 3% for poultry, pigs, and cattle. Nowadays, aquaculture produces almost half of the aquatic products for human consumption, and we have to remember that most of these products come from Asia. The increase in aquatic production is mainly due to aquaculture, with almost 90% increase in the last years, compared to a decrease of the products coming from fisheries by almost 3%. Even though in Europe we like to think broad, we have to remember that the vast majority of this aquaculture production comes from Asia, as we mentioned, and is due to the grass carp, the shrimps, and afterwards the Nile tilapia that is also nowadays farmed in different parts of the world, like Africa, even in South America, the oysters, and the Atlantic salmon. These are the 5 major species that are cultivated nowadays in the entire world. As we can also see, these species present different starting points for breeding, with the first one being the Atlantic salmon from the Norwegian industry some 50 years back in the 70s. And it was followed by the capped oysters some 15 years afterwards, the Nile tilapia, thanks to the gift strains and the efforts that have been started in Asia and Africa, and the white-leg shrimp, and a few years back, also the grass carp and different carp species. This is a nice figure which shows that compared to livestock and farm animals like goats, sheep, and cattle, pigs, marine animals have started to be domesticated not even a century back. So we see on the table on the right that most of the domesticated land plants and animals have, for the plants, we know from historical records that most of the plants have started their breeding efforts in the last 2,000 years, and the big efforts for the land animals started a couple of centuries back. But for the animals and plants of marine and freshwater origin, we see that these efforts, the major efforts, started in the last 30, 40 years, with a peak in the last 10, 15 years. We have to remember that this is a publication some 15 years back already. But at this point, we also should remember that compared to the livestock, the fish, more specifically, present a major advantage, which is their high fecundity. So instead of having a few offspring per generation in poultry, bovine, ovine, and all the other farm animals, at the limit we may have few dozens per year. In fish, we face the fact of the high fecundity. So in Nile tilapia, we may have 1,000 eggs per spawn, and in the Pacific oyster, these numbers can grow up to 100 million eggs per spawn. Now, for the genetic markers, this is a very general slide that shows that the ideal genetic marker, we all know that in order to be included in our analysis, it has to be polymorphic, it should be simple and have a cheap application into our efforts. We should be able to interpret easily the results, also exchange data easily between labs and groups, require small quantity and not very high quality samples. And in the last years, we also face this challenge to be able to automatize them, to be able to run them on the last generation, let's say, sequencing machines and so on. There is a series of DNA-based methods that have been developed in the last 30 years. But of course, in this presentation, we don't have the time to go into details and talk about all these different methods. And of course, we're going to talk mostly for the three types of markers that are currently used into the aquaculture genetics and genomics. The microsatellites are repetitive DNA sequences that are ubiquitous components of the eukaryotic genomes, and they can be up to 66%, so 2/3 of the genome sometimes in few species. They are tandemly repeated DNA sequences of 2, 3, 4, 5, or 6 nucleotide microsatellites that are tandemly repeated into the neighboring regions of the genome. And in the case of mini satellites, these markers have more interest in the human genomic studies, and it concerns usually 40 to 100 base pairs repeats. Depending on their structure, then can be pure, compound, and interrupted. So here we have a pure microsatellite of a CA that has been repeated 10, 12 times. Compound microsatellites, it's a dinucleotide that it's a CA and a GA that is tandemly repeated or interrupted. In fish, the most commonly repeats are GA or CA for dinucleotides, and for the tetranucleotides is GATA and GACA. In general, microsatellites are most commonly found in the non-coding regions of the DNA, but they are also found in relatively high rates into the intronic regions, so the regions that are not transcribed into the genes. They are condominant markers with Mendelian inheritance, and this is a very powerful attribute for their use, because we can theoretically identify lots of alleles into a population. And this is a very important attribute that made the microsatellites a very powerful tool, for example, for paternity issues in humans, in which we have developed specific multiplexes in order to be able to make a correct parentage assignment of every individual based on a set of 15 to 20 loci of microsatellites. The ESTs, the express sequence tags, are developed and are shown through RNA sequencing. So that means that we have to use fresh tissues, we have to follow certain protocols in order to be able not to lose them, because the RNA degrades very fast. And thanks to their poly-A tail, we can take them out of the RNA extract. This is the transcripts, and afterwards to study them. And this has revolutionized the domain of the expression genetics and expression genomics nowadays, being able to study the transcriptome of the fish species and all eukaryotic organisms in general. The oligonucleotide and cDNA arrays have been developed based on the knowledge that we could have from the polymorphism that we identified into these RNAs. And afterwards, we were able to spot probes in this kind of arrays. You can see that are quite small arrays. This is a match, so you can imagine that is arrays that are some 2 by 4 centimeters wide. And with these arrays, we were able to see which genes are expressed at a certain period of the life of the organism. So depending of the color, if it's green, that means it is expressed. If it's red, it's not expressed, and so on. We can monitor the expression of genes through different time points of the organism. And last, it's of course a single nucleotide polymorphism. It's simply decoding the letters of the genetic code. Just to remind you that the DNA is composed of only 4 letters, so only 4 nucleotides: the adenine, the cytosine, the thymine, and the guanine. And these letters are able to code for 20, 30, and more thousands of different products, different proteins in the organism. And this is the causative agent of every trait, or most of the traits that we study in the fish species or in the marine organisms in general, in aquaculture. So, if we're going to compare the power, let's say, of all these markers that have been developed in the last 30 years, we see that the mitochondrial DNA that I prefer not to show has also some power, but it has an inheritance that goes from the maternal parent to the offspring. The microsatellites have multiple number of alleles, so they have a high polymorphism. And the single nucleotide polymorphism, the SNPs, even though they have only 2 alleles, but they can go up to 4, because we use a big number of these SNPs. We can also have a high power of analytical study for the analytical studies. What is very important to know is what exactly we want to study, and depending on the question at the beginning, we may come up to the answer of how many markers do we need finally. If it's about studying the phylogeny, let's say, of some species, we could simply use Sanger sequencing on few genes and have our reply. But as we are aiming to more detailed studies and questions like population structure, QTL mapping, pedigree mapping, association mapping, population genomic scans, we need more and more markers that can be from few tens to few hundreds and thousands of markers. And in that case, it's the SNPs that are going to give us all these markers. A protocol, a methodology that has been developed is the RAD technique, but the advent of high-throughput sequencing platforms was something that revolutionized the genetic studies in the last 20 years. Starting from big machines that occupied half of a lab, we nowadays have the possibility of benchtop-like machines that are easily transported and can give us in few days and few hours, sometimes if it's about bacteria, an amazing amount of genetic data that can lead us to genome assemblies. Of course, there are trivial issues like the long or short reads, the speed, the cost, and the quality of the reads, because this is something that we have to bear in mind. Some technologies, some methodologies produce reads that can be short but can be very accurate, and others can produce longer reads but instead contain a high percentage of errors that should be corrected. So nowadays we have in our arsenal machines that produce short reads like the Illumina type machines, and with the new generation of the NovaSeq, we are able to produce more than 3,150,000,000 base pair reads per day. So this is translated to 450 gigabases per day. Just to give you an order of magnitude, the human genome is around 4 gigabase pairs, so we can produce the amount of 100 human genomes per day, and a normal fish is around 1 gigabase pairs or lower, so 450 fish genomes per day with these technologies. We also have technologies that produce longer reads, like 13 kbs, 13,000 base pairs per day reads. So it accumulates to 20 gigabase pairs per day, 20 genomes of fish per day, and others like the Oxford nanopore, which can produce longer reads up to 200 kb and 7 gigabase per day. As I told you, in the last 15 years, since the description of the restriction site associated DNA sequencing, the RAD methodology in 2018, we have developed, I mean, the community of the geneticists or in molecular biology, more than 15 different methodologies, GBS or DDRAD or 2BRAD, that nowadays are very frequent in their use in the study of marine organisms. And this is a more or less old publication that shows that there is an increasing number of studies year by year with all these different restriction site association DNA sequencing methodologies. It is simply methodologies that use restriction enzymes to cut the DNA in different pieces, and afterwards mainly with Illumina sequencers to be able to have multiple copies of 150, 300 base pairs fragments. And there the challenge is to align them. Just a short parenthesis that the restriction enzymes are microbial products that are used for defense in the microbes, in bacteria, and they are palindromes that recognize specific sites in the genome, and they can recognize 4 base pairs or 6 or 8, and they can have blunt ends, so they can have abrupt cuts in the genome, or sticky ends like the Tetris motifs, and their use is quite abundant in the last years. And of course, we are able in silico to evaluate what effort we need into the sequencing process in order to choose the best enzyme to cut our genome. So in the case, let's say, of a 6-base cutter, we have a pattern that should be observed nearly theoretically every 4,000 base pairs. So if we have a fish genome of approximately 1 gigabase pair, we are expecting to have 250,000 restriction sites and fragments afterwards into our data set to study. And as I told you, the RAD-seq techniques are used more and more in the last years, but of course, there are advantages that are associated with the application of these techniques and other disadvantages. and the main disadvantages being the complex library preparation, that it's labor intensive, we need time, and afterwards we can see that also in the last 10 years, these methodologies have been used in a variety of studies in salmon, rainbow trout, and recently to carps and turbot and other fish species. So it's a technique that is being used more and more to study growth, sex determination, disease resistance, and so on. A very crucial point, stepping from the, let's say, first genetic methodologies like microsatellites, in which we were able to run few softwares of population genetics or parentage assignment. Nowadays, with these millions and billions of sequences that we have from these methodologies, we absolutely need bioinformatics support. And not only that, we need high-performing computers, capacities that are not in every lab, that are not sometimes in every country. So we need help from partners that can be in a neighboring lab or in another research center or in another country. We can see that if we pile up all the sequences, we can identify different SNPs. And if we consider, for example, this is the chromosome of the stickleback, we can identify where exactly we can position, we can locate all these SNPs onto this chromosome. And if we put all these chromosomes together, like the 21 chromosomes of the stickleback, we can see regions in which we have more and less SNPs, because SNPs, theoretically, there might be equal distributed into the genome, but in practice, they are not. We are able also to do multiplexes, so to use into these methodologies different individuals that are dyed with different colors, let's say, and afterwards be able to make a catalog and identify this catalog and identify into the different stacks, the different haplotypes of the specimens that we have used. Okay, it's not a black hole, it's not magic, but it needs specialized personnel to be able to run all this analysis before getting to the final data set. What we also see in the last years is the more and more frequent use of the SNP arrays. In order to be able to design and order this kind of tools, we should first identify a few hundreds of thousands or even millions of markers per species, and after the different filtering steps, to choose the best ones and spot them on this array. These arrays are designed into different in different formats, like 96 or 384 formats for 96 or 384 samples respectively, and can accommodate a great number of SNPs. And into this study, we should acknowledge that the salmon has played a crucial role bridging the technology transfer from the livestock, because all these technologies and methodologies have been first developed in poultry, the bovine and ovine and pork industry, into the Mediterranean, and not only species. All the marine species have taken this advantage of the work that has been carried out in salmon and rainbow trout. And nowadays, depending on the density of the markers, the community has developed low density, medium density markers that go up to 60, 90,000 markers. And we are able also to go for 250 or 690,000 SNP markers in catfish. Of course, in the livestock industry, there are SNP arrays that comprise more than 1 million markers, but it is yet to come if it is judged important also in the aquaculture industry. At this point, just a few slides to show you what has been done into the PerformFISH project with collaboration with the Medate project, in which we used pools of individuals based on the pool-seq methodology from 45 sea bream and sea bass populations. So in total, we studied more than 1,000 fish, both farmed and wild specimens, to identify SNPs. So we have populations that were gathered from all around the Mediterranean, and unfortunately we had few populations from the Atlantic. And for these populations, we made pools of samples. So 25 DNAs from the samples of one population were pulled together and sequenced. So we couldn't have individual information, but we had population information of SNP frequency and presence, of course. So in the European sea bass, we were able to identify some 17 million markers, and in the gillhead sea bream, some 34 million markers. You can see their distribution on the chromosomes, on the linkage groups, with the red parts being having more frequently SNPs. And afterwards, based on the SNPs that we identified, and on other SNPs that we knew in advance that were associated with relevant production traits, and also with SNPs that were used into other platforms like the Genesee arrays developed in France, we were able to design and validate the MedFISH array, which is both for bass and bream. So it's 30,000 SNPs for sea bass and 30,000 SNPs for sea bream to be used. Compared to the RAD-seq methodologies, the SNP arrays are more straightforward with a less bioinformatic labor. They present a high accuracy and high repeatability compared to the RAD-seq. Unfortunately, they are fixed. It's only the SNPs that we have spotted onto the array that we can study, and they have, of course, a bigger cost than the RAD-seq. The last part goes to the reasons why we want all these markers, and of course, is to study the genetic diversity of aquaculture stocks, because we may think that the aquaculture stocks are well genetically characterized, but it's not always the case. And we have to use these markers in order to differentiate strains and subspecies sometimes. And also, we know that the domesticated stocks start to accumulate SNPs, or the frequencies of the SNPs change over time due to selection. And this can make them different from the wild counterparts. So it's a nice example to study escapees. To this extent, microsatellites and SNPs are the best markers to use. And also in the case for parentage assignment and reproductive contribution, because we need to have high and reliable assignment rates. as the cost for rearing and phenotyping candidates is high, so we need to know the offsprings of the broodstock that we have used. In that case, the microsatellites provide a great help. In most cases, 10 to 15 microsatellites provide a high assignment rate. And in the case of SNPs, because they are biallelic, so they present only only 2 alleles, we need an estimated 3 to 7 times bigger number of SNPs to have the same analytical power. We need them to monitor inbreeding, which is the mating between sibs. And inbreeding, we know that generation after generation, that can lead to inbreeding depression, which is reduced reproductive capacity, lower FCR, increased frequency of deformities, and so on. Sometimes when doing in-house selection, we choose the best performing animals, but in most of the cases, these animals are sibs. So we risk into the next generations to have sibs, to have brothers and sisters, cousins, and after generations, this will lead to the inbreeding depression. So we have to avoid that by using genetic tools and to monitor the inbreeding. We also use genetic markers to monitor sex, because in lots of species, we know that some sexes, specimens of one sex grows faster, like males in tilapias or females in flatfish and sea bass. So if we can identify markers that can skew this percentage, we can afterwards choose and cross these animals to have a bigger percentage of the sex that grows faster. In the last years, we are also interested in genetics for feeding, studying and choosing genes that provide adaptation to different feeds, lipogenesis and enzyme synthesis. And what is also very interesting in the last years is to study metabiomes. And from one hand, we have the microbial communities that exist in the digestive system, the digestive tract of the fish, because the fish fauna and flora that exist in the gut plays an important role in the welfare and the FCR, and also metagenomic approaches that are referred to the fish cage and the tank environment that can play also an important role. Not to forget the very interesting input in the last 10 years of the epigenetics. And into this, we include all the inherited changes in gene expression that are not into our DNA sequence. So it's factors that, although not of genetic origin, affect gene expression through different mechanisms. And these factors can be passed, can pass their imprint to the future generations, even if the original stimulus no longer exists. The very interesting for the epigenetic mechanism is that they don't have a Mendelian type. It's not easy to study them, and depend mostly on the DNA methylation. And we have seen that in the last years, the effects of epigenetics and the stimuli that occur during early development are very, very important, and they can change a lot the domestication process. Of course, we're using markers for the marker-assisted selection, and it would be ideal if we can find a marker that could be used afterwards in order to choose for better growth, for better productivity, for better FCR, for higher disease resistance. Even though nowadays our efforts are focused on what we call genomic selection, in which rather than seeking to identify individual loci and do a marker-assisted selection, we use thousands of SNPs to screen the genome of the specimens and predict performance. In the next years, with the advent of the whole genome sequencing and the wealth of the data that are going to arrive, we think that it will be more justified to go for low coverage whole genome sequencing. So instead of having SNP arrays that may have a bias and a certain bias and skew somehow the genomic prediction with issues to the linkage disequilibrium and so on, the scientists predict that in few years, instead of using even arrays, we're going to go to low coverage whole genome sequencing. Last, we should remember the reason why a few years back, two scientists had the Nobel Prize for this CRISPR-Cas9 methodologies, which is in fact this cut and paste techniques that can bring genes from other organisms or change the frequency of already present genes into the genome. And with these techniques, nowadays, for experimental reasons, we have attained lice susceptibility Atlantic salmon and lice-resistant coho salmon, and they have been used also in livestock breeding programs. But for the time being, these methodologies are still considered as GMOs producing, so it's considered transgenesis, at least in Europe. We don't know what will happen in few years. I think that in time, we may have few questions, if there are, before getting to the next presentation. Otherwise, in order to respect the 5 minutes remaining till Mark starts his presentation, I don't see that we have questions. I'm just going to show you the latest map on gene editing regulations. So we see that depending on the region of the world, we can see that this is, of course, for plants and how what's the perception of the policymakers and our rulers, let's say, our governments. So in fact, in New Zealand and in most of Europe, the genome edited crops are regulated as GMOs, whereas in other parts of the world, like in US, the Canada and Southern America, we see that the same crops are not regulated as GMOs. So we are living in very exciting times. We are expecting lots of things to happen, and we have to see what will happen in the future. So still no questions, so I will stop sharing. Mark?
Okay, so I will start sharing. Is it okay? Do you see it properly?
Yeah, Mark, it's excellent.
Mm-hmm.
Just to wait a couple of minutes,
Yeah.
if you don't mind, to be exactly on time. Let's start.
In case some people come specially for me, yes, that would be.
Yeah, I think it's very probable. Huh? You are a star.
Yeah. I think you made a very nice presentation of all these new technologies.
Yeah, it's not easy to compile everything in 40 minutes, but okay.
We have a question. So maybe you want to answer, Costas?
If we're able to determine the type of sex in spawned eggs through genetics, to my knowledge, there are very few species in which we have a well-known sex determination mechanism with chromosomes or chromosomal regions or genes. So in this case, like if I remember well, a few species that make the caviar, if I don't remember, maybe we are able, but in our species, I mean in the species sea bass, sea bream, salmon, rainbow trout, we are not able, even in later stages before.
I think in the salmonids there is a diagnostic marker. So
there is.
Salmonids, yes, they have a chromosomal sex determination, and the sex determinant is known. So
yes.
There are genotyping assays in salmonids. Maybe to precise a little bit more, spawned eggs, no, you can't, because spawned eggs, they are not fertilized, and to know the sex, you need to know the male and the female. So once they are fertilized and they start developing, and you have the genome of the future individual in the egg, then in some species you can, but in some species you can't. I mean, in sea bream, as they change sex during their life, it makes no sense to say that you can do that. And in sea bass, it's complicated because there are many genes which are influencing sex. So we can, if you have previous genetic information on that specific population, you can give a tendency that this or that animal would be more or less female-like or male-like, but in the end, this will depend a lot on the environment in which they develop.
Mm-hmm. Okay, I think we can go to your presentation. There is another question that I think we will be able to reply in the session of question and answers afterwards, in order not to delay your presentation, Mark. Please continue.
Okay. So I'm doing a kind of back to basics, the basis and constraints of selective breeding in aquaculture. I think it's interesting for people who want to start a breeding program, but it's interesting for scientists also, because we as scientists, we are very much focused on discovering new mechanisms and fancy things. But these, when we are talking about selective breeding for aquaculture, we are talking about production, we are talking about improving populations. And this has many specific constraints which have some consequences on the way to translate the results of research into something that is effective for production. So if I start with what is selective breeding, you can define it quite simply. It's a way to improve the genetic level of a selected population from generation to generation. And to do that, you choose as broodstock within this population the animals which have the best genetic value for the breeding objective that you have, and you use these animals to breed the next generation. That's quite simple. So it's simple. You have a number of parents, you reproduce them. With that, you produce a number of fish that you call selection candidates. And among these selection candidates, you choose the best parents, the best animals to be the parents for the next generation. And this goes from generation to generation. This part of the breeding program is what we call the breeding nucleus. So it's really where selection takes place from generation to generation. And from that, you have to output some juveniles for production, because what you need in the end is to have improved animal for the aquaculture production. So, okay, this is quite simple. We know also that selective breeding can be very efficient. If you look at this, so it's data from animals, not fish, but poultry, but aquaculture and poultry farming have many similarities. On the left, you have the result of an experiment where I started to say fish, but no, they are not fish. They are chickens. So chickens from a 1957 strain were compared to a 2001 strain. So the 2001 was selected from the 1957, and you see that after this selection, there is really a huge difference between the two strains. The body weight has been multiplied by 4 to 5. Carcass yield has increased by 25%, and breast yield has almost doubled. So this means that this is really a powerful tool. You can really completely change the animal that you are rearing, make it completely different using this selective breeding. Very striking example is on the right, where you have selective breeding of turkey, which was followed over 28 generations, and which was targeted to body weight. So the body weight is measured as 16 weeks, and we see the increase from generation to generation. And what you can see is that in the first generation, the body weight was less than 500 grams at 16 weeks. And in the 28th generation, it's around 5 kilos. So it has been multiplied by 10 in 28 generations. This is to give you really an example of how powerful selective breeding can be. We know it works in fish also. If we use the same trait, growth, here are the results of a few documented selective breeding programs where growth was targeted on tilapia, on trout, on salmon. And I standardized all of them to a level of 100 at the beginning of the breeding program. And what you can see is that in a few years, it's perfectly feasible in fish to double the growth rate of the animals with selective breeding. So this looks simple, this is efficient, so we have to do it to improve the quality and the efficiency of our fish farm. But okay, this looks very simple, but how simple is that in reality? So we said that we wanted to choose the best selection candidates to select the future parents. But what means the best? Best for what? Well, the traits that you want to improve can be very, can be many traits. For example, it can be growth, it can be disease resistance, it can be quality of the fish, efficiency like fillet yield or FCR. And basically, you can improve any trait that you can measure. If you can measure a trait reliably, you can improve it. So you have to choose the traits you want to improve. But a specificity of these traits that we are interested in production is that these are what we call quantitative traits. So the phenotype, the performance of the fish, is a numeric value. There is no two categories. There is no good fish and bad fish. Even for phenotypes performance that look like good and bad. I mean, if you have disease resistance, okay, in the end, the fish is dead or alive. You could say it's a binary performance, but in fact, in reality, it's not. It's the realization is binary, but the trait itself is continuous. Fish can be more or less resistant, and depending on the conditions, they will be live or dead. So it's even these simple, apparently binary traits, they are these quantitative traits. And if I take, and these quantitative traits, in fact, we can statistically treat them, and they are composed of different types of performance. So if I look at the phenotype, the performance, it's the sum of the mean of the population of a genotype effect and of an effect of environment. For example, if I have a sea bass which has a body weight at 2 years of 440 grams, well, it's from a population with 420 grams mean weight, and it can have a genetic value of plus 30 grams and an environmental value of minus 10 grams. That makes this 440. And the main characteristic of these quantitative traits is that they have, in general, a polygenic genetic basis, which means that there are many genes that are implied. And when I say many, it can be hundreds, it can be thousands, it can be really a high number of genes. And so for most traits, you cannot say, for example, I have the gene of growth. This fish with this gene is the best grower. No, it doesn't work, because growth is influenced by thousands of genes. So in the end, what you want is to have the fish which have the best genetic value for the traits that you are interested in. And why do you want that? Because this genetic value is transmitted to the next generation. So if you use the fish with the best genetic value, then your next generation will be improved for this trait. But then comes the question, which fish has the best genetic value? If I have some sea bass in a tank, okay, I want to sort them on the genetic value, but what I can measure on this fish is the phenotype, which is the mean of the population, the genetic value, and the environmental value. And how do I identify the ones with the best genetic value, so this G is also what we call the breeding value of an animal, but it's not that easy. And one thing that we use to try to access that is we use what we call heritability. Heritability, it's the proportion of the variation in the phenotype that you see in the population that can be attributed to genetic variation. This heritability, it varies between 0 and 1. And what we can say is that if heritability is high, a lot of the phenotypic variation is due to genetic variation. And in this case, phenotype is a good predictor of genotype. But if heritability is low, only a small proportion of the variation in the phenotype that you see is due to genetics. And then phenotype is a poor predictor of genotype. Heritability can be estimated from measurements of phenotype in quite a high number, usually 1,000, 2,000 individuals from many known families. So if you have a known family structure and phenotypes on many individuals, you can estimate the heritability of a trait. And why is it important to know the heritability of a trait? Because from this heritability depends your capacity to identify the fish with a good genetic value and your capacity to have an efficient selection. If I look at the effect of heritability on the relation between phenotype and genotype, so and phenotype and the breeding value, if the heritability is 0, it means that 0% of the variation of phenotype is due to genotype. So it means that basically I have fish with different phenotypes, but they have all the same genotypic value. So if I select the 10% best fish, they will have 0 genetic value. They will be exactly have the same genetic value as the other fish. If the heritability is a bit higher, 0.25, there is some correlation between phenotype and genotype. And if I choose the 10% best fish, the average genetic value will be a bit over the average of the population. And as heritability increases, the genetic value of the selected fish will also increase. And so my selection will be more and more efficient because if I select the best fish, their genetic value will be higher if the heritability is higher. If heritability is 1, all the variation of phenotype that I see is due to genetic variation. And then selecting on phenotype is equivalent to selecting on genotype. So selecting animals with the best breeding value for G, for the trait, is easier when heritability is high. This is why we want to estimate heritability of the traits. And in PerformFISH, we have estimated the heritability of many traits in our CBAS and CBOOM. Then we come to a second simple questions. Okay, we want to choose the best candidates to select the parents, but how many parents do we need? Well, when we do selective breeding, I said the breeding nucleus, the population on which we select, is closed. It means that the parents come from the offspring of the preceding generations. And this has 2 consequences. If the population is closed, sooner or later, you will have to mate individuals which are related to each other. And if you mate individuals which are related to each other, then inbreeding increases. And the second consequence is that as the population size is not infinite, it's finite, at each generation, some alleles will be lost by a phenomenon that we call genetic drift. Imagine that you have a population of only 10 fish, only 1 fish carries a specific allele. If you don't select this fish, this allele will be lost. And if it's lost, it's lost. It will not come back from nowhere. I mean, it's really variability, genetic variability that is lost. So to try to counteract this, there is basically only one solution. You need a high effective number of broodstock at each generation to keep inbreeding low and to conserve genetic variability. If we look at the effect of this effective population size on the loss of genetic variability and on the inbreeding, I have made here 2 simple graphs where you see on the left how much of the initial genetic variability remains in your stock after a certain number of generations, up to 20, with different effective population size. And you can see that if you have an effective population size of 10, after 20 generations, you will have lost more than 60% of the initial genetic variation. So a huge part. If you want, after 20 generations, to still have 90% of the initial genetic variation that was available in your population, you need to have an effective population size of 100. And for the inbreeding coefficient, it's the, I would say, the exact reverse. If you have a small effective population size, inbreeding coefficient will increase very fast. You have to know, so here you have the inbreeding coefficient, which can be 1 at the maximum. 1 is a clonal population. So if you see that if you use NE of 10, after 20 generations, you're more than halfway to a clonal population. So you have lost a lot, you have lost a lot of variability, but also you have individuals which are very much related to each other. And here, the general recommendation in the breeding programs is to keep the increase of inbreeding, which we call delta F, to less than 0.5% per generation. And this implies that if you want to do that, you have to have an effective population size which is more than 100. And this is very important because if you want your breeding program to be sustainable, if you don't want to have to input new blood in the breeding program after some time, you have to stick to these numbers. And this is not negotiable. There is no way to overcome that. And if you don't do it, at some time you will get into problems with inbreeding with little genetic variability, and you will have to bring in this new blood. But by bringing in new blood, you will also lose the effect of selection that you have gained over generation. So it's not really what you want to do. But then we go to another problem. Is this effective population size equal to the number of parent fish that you keep as broodstock? Well, it equals this number of parent fish under a certain number of conditions. All the parents have the same average offspring production. The number of males and females is the same. All parents are reproduced, and there is no selection. In reality, you see that these conditions never apply. So what happens if they don't apply? First, let's say I have 20 females and 25 males, so a number of parents of 50. If I have all parents which have the same average offspring production, which means that offspring number per parent is random, I will have an effective size of 50. Okay, NP is equal to NE. But if it's unbalanced, something that happens very easily, especially if you use mass spawnings or if you have mortalities or things like that, you can have, with the same number of 50 parents, an effective population size of only 70. On the contrary, if you make sure that you have exactly the same number of offspring from each parent, and these offspring are used to produce the next generation, you can have an effective population size which is larger than the number of parents. But this has a high cost in terms of knowledge and organization, because you need to know for each offspring which are the parents, and you need to be able to make sure that in the next generation, each parent gives exactly the same number of future parents. So that's quite complicated. Now, the effect of sex ratio. If I have 100 parents, if I have 50 males and 50 females, the effective population size is 100. But if I have only 20 females and 80 males for the same total number, then the effective population size is 64. And if it's very much unbalanced, like 2 females and 98 males, the effective population size for 100 parents will be only 7.8. So you can see you can have a drastic reduction. So for this, you need to try to equalize the number of males and females contributing to the offspring. Now, another practical problem in a hatchery is I have my parents in the breeding tanks, but do they all contribute to the next generation? Do they all breed the day when I want them to breed? And the answer is generally no. Some will contribute and some on that day will not produce eggs, will not fertilize the eggs, etc. So for example, if only 30% of the males and females participate on a given day, well, the effective population size is just 30% of the number of parents that you have stocked. And then selection. Typically, so the calculation is a bit complicated, but typically if you have 5 to 10% selection pressure on a trait, what does selection pressure? If the trait you select has some heritability, it will favor families which are good for that trait. And it will create unbalanced family size. And typically 5 to 10% selection pressure on a trait which has a decent heritability will divide effective population size by 2. So now let's get into the worst case scenario. Okay, I want to have an effective size of 100. I do a selection pressure of 10%. So this translates into 200 that I need. Only 50% of the fish reproduce on a given day, 400. My sex ratio is not balanced, 625. And I have unbalanced families. And okay, I can need 1,800 fish in my broodstock to be able to have an NE of 100. So you see that you have many points that you need to control to have this effective population size, which again is not negotiable. So now I know how many parents I need, but how many selection candidates do I have to breed and measure to select these parents? Well, this number of selection candidates conditions selection intensity and genetic gain. We have what we call the breeder's equation, where the genetic gain in one generation is function of I, the selection intensity, and something else that I will not talk about in details now, but which depends on heritability and how you evaluate the breeding value of the animals. Now let's concentrate on I, the selection intensity. Selection intensity is a number that depends on the selection pressure, which is the ratio of the number of parents that you will select divided by the number of candidates. So if you select 50% of the parents, then the selection intensity will be 0.8. If you select 20%, it will be 1.4, etc., etc. I remind you that number of parents is fixed because effective number is not negotiable. So let's say that I need 300 parents to have my NE of 100. It means that I'm not too bad at a sex ratio, a number of parents that breed, equilibrium of family size, etc. So if I want to increase genetic gain, I want to increase I, so selection intensity. So I want to have a higher selection pressure. So if I have a selection pressure of 10%, I need to have my 300, I need to have 3,000 candidates evaluated. And if I want to have 1 per 1,000, because I want to be very fast, I need to evaluate 30,000 individuals. So quite a lot. But note that I is not linear with selection pressure, though. So by going from 3,000 to 30,000, you'd only double selection intensity, and thus you only double gain. So you multiply your effort by 10, but you only double gain. So this is something that you have to weigh when you design a selective breeding program. And this is very important because remember that all candidates have to be evaluated. If you want to choose the fish, you have to measure the fish to know which ones you want to choose. So measuring 3,000 fish or measuring 30,000 is not the same cost, not at all. Then one more thing is, okay, we want to choose the best fish, but how to choose the fish? Well, the simplest is when you have one trait, which is measured on all live candidates. It's what we call mass selection or individual selection, which works very well when you have a heritable trait with, say, heritability of more than 20%, and which is easy to measure. Typically, selection for growth. Selection for growth is easy. You measure the fish. Generally, heritability of growth is 0.2, 0.3, 0.4, 0.5, something quite nice. It's easy to measure the size of a fish, so you can select very easily. And measuring the size of a fish does not kill the fish. So you can, after measuring it, you can keep it as a future broodstock. There is no problem. But okay, it's not always the only thing we want to do. What happens if we want to select not on a single trait, but on a combination of traits? What if the trait is not measurable on the candidates? And what if the trait is not heritable enough? Well, first, for selecting on a combination of traits, you have to define your breeding goal. Your breeding goal is a linear combination of the different traits you are interested in. What this means is that it is always a compromise. If you want to select on many traits, you cannot get the maximum gain on all the traits because you will have to share the weight of these traits in the breeding goal. Ideally, the weight of this breeding goal should be derived from a production model such that this breeding goal maximizes an economic gain. But having this model working in all conditions is not always very simple. And very often what we use is what we call desired gains. So the breeder has an idea of which traits he wants to put the focus on, and he will choose himself the weights to put on each of the traits. And then once you have this breeding goal, you have to build a selection index where you will make another index which is dependent on the traits that you measure on the fish, but also on maybe other traits that you cannot measure on the fish, and but that have some impact on the traits you are interested in. And these coefficients, which are not the same as the first ones, they depend on the coefficients in the breeding goal, but also on the heritability and the genetic correlation of the different traits. So it's not that simple to select for a combination of traits. What if I want to select on a trait which I cannot measure on the breeding candidates? For example, disease resistance, fillet yield, feed efficiency. If I fillet a fish, it's not a good candidate for selection after because it's dead. Okay. If I want to measure individual feed efficiency, I need to have each fish in an individual system. I cannot do that on 30,000 breeding candidates. It's not possible. But I can use an indirect trait. For example, in PerformFISH, we have shown that the individual feed efficiency of sea bream in individual aquaria is related to the fat meter measurements in adults. So it has a genetic correlation, which is not very strong, but a negative genetic correlation. So if I select to lower the fillet fat content in the adults, I will increase to some extent individual feed efficiency. That's one way to do. The other option is to use family links that we call pedigree selection or genomic resemblance, genomic selection, to do seed selection. So we have some challenged fish for which we have phenotype and pedigree or SNP genotypes and phenotype, and selection candidate, and we measure them for the difficult traits. And we have selection candidates for which we have only either the pedigree or the SNP genotypes. Pedigree can be obtained with separate rearing of the families or with parentage assignment. And SNP genotypes are typically obtained with a SNP chip. And if I do this, I can derive the potential breeding value of the candidates from the measurements that I've done on the challenge fish through the genetic links. And for example, in C-bream, we could see that we could have quite a good accuracy of selection, better accuracy with genomic selection than with pedigree selection for feed efficiency using these family links. So it makes possible to select many individuals knowing only genetic information if you have a reference population where you have genetic and phenotypic information. And then if the trait is not heritable enough, you can do the same, use family links or genomic resemblance. And instead of having only the phenotype of the candidates, if you have their phenotype plus pedigree or SNP genotypes, in fact, you will use information that you have on related individuals among the candidates to have a more precise evaluation of each candidate. And if I have an example with where I use only the phenotype of the candidates, you see that the average genetic breeding value is not very much above 0. But if I use pedigree or genomic information, I can have a higher correlation between this index and the breeding value, and then have a more efficient selection. So I improve the accuracy of my breeding values. Then, okay, I've managed my nucleus. Then I need selected juveniles for production. So where do I get them from? Well, it depends. Basically, this is what we call the multiplication step. It depends on the size of the production. And I can directly use selected parents to produce the offspring. With that, I have the highest genetic level, but a low number of offspring. If I have a huge production system to fill after that. I can also replace my hatchery broodstock with selected parents, but this is only progressive, so I will have a delay in having the genetic impact on the production. If I need even more broodstock, I can replace my hatchery broodstock with unselected candidates. So these are the ones which are below the genetic level of those which are used as a replacement in the nucleus. So they are still improved, but less improved, so it gives even more delay. Or I can produce multiplication broodstock with elite parents. So in this case, I choose the very, very best parents and made them to have the future broodstock that I will use for the production. It gives 1 generation delay, but I can do very strong selection of these elite parents. So this can somehow balance things. And I can also use this to diversify markets. For example, if I have a marker that identifies fish which are resistant to a specific disease, I can produce specific broodstock for a specific market that will be resistant only to that disease. So you see here also we have many options. So in the end, that was very simple, yes. In the end, it's not that simple. But so how does it look like in the end? I have my effective parents, they need to be more than 100. I have my candidates. I have the phenotypes of these candidates. I can have pedigree or genotypes to improve the selection of the parents. I can have some fish that are challenged and pedigreed or genotyped to be able to have breeding values for traits that I cannot measure on the candidates themselves. And what remains important is that phenotype is the key. I need to be able to measure the traits that I want to select accurately enough on a large number of fish. I need to phenotype or genotype many animals. So I need efficient and low-cost phenotyping and genotyping. The effective number of breeders is something that is not negotiable and conditions all other numbers. So you have a lot of work to do to be sure that the number of parents that you have is high enough to have this number of effective parents that you need, and that the difference between the two is as small as possible by controlling everything that I've shown. Because if I want to have, if I have 100 effective parents and want a pressure of 10%, I will need to measure 1,000 animals. But if I need 300 parents to get the 100, I will need to measure 3,000. So this has really high consequences. It's important to note that it's a long-term process, because the unit scale is the generation, and that pedigree or SNP information can improve precision and authorize little traits, but these have a cost. But selective breeding is really versatile. I mean, any trait can be improved by selective breeding, provided that you can phenotype it. This is very important. The key is the phenotype. And a kind of meme that is very popular among breeders is that in the era of genomics, phenotype is king. If you don't have a good phenotype, you cannot do good selection, even with the best genetic and genomic tools. The other thing which is very important is that genetic gain accumulates over generation. So even if you gain little by little, what is gained is gained. And we could see that there is virtually no limit. You could see, for example, in Turkey, the body weight at a given age could be multiplied by 10. Who would think it is possible in the beginning? No one, but yeah, it is possible. So there are some physiological limits, of course. If I want to select for fillet yield, I cannot have a fish which is composed only of fillet. It's not possible. So at some point, there are some limits, but these limits, very often we think there are more limits than there are. Here, for example, you can see, so it's not animal, it's a plant. It's a breeding program on oil content of corn, which is done by the University of Illinois since 1896. And you can see that over more than 100 years, the genetic gain has remained linear. So it means that, yes, you don't exhaust genetic variation that fast. One other thing which is very important is that the breeding goal can evolve over time. If you look at this graph, you can see that after 50 generations, they decided to select for lowered oil content. Okay, if you select then for lowered oil content, the oil content decreases. And if after 10 generations, you change again your breeding objective to, okay, I still want to increase it, it works also exactly the same with the same slope as if you had continued the initial breeding. So as you are supposed not to be completely stupid and to think a bit on the breeding program, your breeding goal may evolve over time, but probably it will not completely change. You will not say, okay, what I've done until now is completely stupid and I want to do the exact opposite. So it's very interesting because you will keep the gains that you have done and you can then change the objective, and this will go smoothly from generation to generation. To do that, knowing genes can be useful in specific cases, but in general, the key, the engine of the breeding program is phenotypes, genetic resemblance between animals, and computing. And with that, this is really what makes this progress from generation to generation, which can reach very high level. And this is why I'm really convinced that selective breeding is and will remain the basis of genetic improvement of fish. So you see, in the beginning, it seems very simple. It's not that simple. There are many constraints with high impact, but okay, in the end, this is very efficient. This can vary over time, and this is why selective breeding is used in all farmed animals, in all farmed plants, because of this very high power in the long term and versatility to have evolving objectives. And I think I'm done with 5 minutes in advance, I think, yeah.
No, it's great, Mark, because we have the time for a couple of questions concerning your presentation. Great talk, thank you. The first one comes from Ivo Monteiro, and he states, if we are too worried about genetic variability lost, if we are concerned too much about the decline in the genetic variability in the stocks, I guess.
Well, I think we are not worried enough. Why do I say that? First, if you look at the real mature breeding programs, for example, Atlantic salmon, the number of broodstock that they use is really important. I mean, it's several hundreds up to more than 1,000 per generation. I think it's not for no reason. The thing is also that if you look with genomic tools at the genetic variability that is lost, now with the high definition genomic tools that we have, we can have a look at how genetic variation is lost over generations in a population, just by looking at the end generation. You don't need to have data in all the generations. You have ways to model how genetic variation was lost over generations. And what we see in the fish breeding programs is that the loss of genetic variation that we see using these genomic tools is more than what we think we would
Can you hear us, Mark? We don't hear you.
Do you hear me now?
No, it's okay.
Okay, so I don't know, I was disconnected for I don't know which reason. So yes, we see that the amount of inbreeding and genetic, so increase of inbreeding and loss of genetic variability is more than what we think when we look at what we know of the number of broodstock which is used and so on. So, okay, for the moment, most of the breeding programs in fish are only a few generations back. I mean, if you look the oldest selection program in sea bass, the oldest sea bass now, there are only 8 generations from the wild stage. So we are really at the beginning. So, okay, when you are in the first 2-3 generations, you can think, oh, okay, I'll see that later. But then if you see that later, you may really get into big problems at a later time. So I think we're not worried enough.
Thank you. Another question before getting to Sarah. Sachi asks if a reduction of the duration between each generation is linked with the increase in whole body mass gain. I mean, when the fish are growing bigger, the generation is
shorter.
Yeah.
So I'm not sure I completely understand this question, I mean, there are two ways to answer to this. First, if you shorten the generation, you increase the genetic gain per year. So this can be interesting to have higher genetic gain. But there is also this link between growth and reproduction. And it's true that in some cases, you see that fast-growing fish will probably mature earlier, so at a younger age than slow-growing fish. So this can increase genetic gain, which is good, but this can also cause problems because usually we don't want to market mature fish. So if this increase in growth causes early maturation, this can be a problem in production. And for example, in salmon, they have always selected against early maturation to avoid that the marketed fish are mature.
Mm-hmm. Okay, thank you, Mark. We have 2-3 more questions for you, but let's let Sarah begins her presentation, and yes, we will attack that on the question and answer session after her presentation.
Okay. Can you see the presentation properly?
Great, Sarah, you can go on.
Okay. So yes, I'm very glad to present some notes about the methods and the importance of parentage assignment and genomic selection in aquaculture species that are relevant topics for those involved in or interested in breeding programs. So, okay. Okay, so let's start with parentage assignment. That is the first part of the presentation. And as you will see during the presentation, parentage assignment and genomic selection are strictly related to each other when we run a breeding program. To start with a brief background, how can we deal with the problem of parentage assignment? So in the past, the first and simpler solution was just avoiding the use of pedigree as in individual selection or mass selection. In this method, the selection of the individuals was based on their own phenotypes. These are cheap methods. They are cheap selection methods. So we don't need individual identification or the maintenance of pedigree records. And also, if the trait of concern is characterized by higher heritability, as happens for growth or morphological traits, we can also expect a rapid improvement in the future generation. And this is what happened in the past, actually, for traits like growth. That was, I mean, the most important trait. But the disadvantages are that we have a high risk of selecting progeny from few parents. We just select animals from few parents producing the best offspring. This increases the risk of inbreeding. And thirdly, what happened is that we have limitation in study genetic variation of the trait of concern. And obviously, as the selection of the individual is based on their own phenotypes, they are not suitable methods for traits requiring the slaughter of the animals. So as we have already seen from the presentation of Mark, if we are interested in traits requiring the slaughter of the animals, like carcass traits or flesh quality traits, fillet-eared, or any kind of challenges to assess disease resistance, for example, we cannot just select the animals we have phenotyped. So what is evident is that we need to control the parental contribution to the next generation. So we need pedigree records, and we have to find a solution to have these pedigree records. We can deal with this problem rearing the different families separately. Yes, it is something that we can do, but rearing each family in a different tank, so I mean 1 tank per family, is difficult. It is space and labor intensive for sure. We need infrastructure, and it's not always possible to increase the number of the tanks we have in the farm. So in this case, we are limited by the number of available tanks. We have to produce as many families as the number of tanks we have. And also, there is the disadvantage that common environmental effects between tanks may alter the heritability estimates of the traits with a great inflation of the estimates. We have estimates of heritability that are not real. So this is not an appropriate solution at the end. Another possible solution is recording the sire and the dam used to produce an animal, then tag the animal to be able to recall its parents. And then we can mix the different families, different individuals from different families in the same tank. But as we work with fish species, we know that animals at hatching are too small to be tagged, so we cannot tag animals at hatching. And again, that's not an appropriate solution. So we have to rear different progeny groups together in the same tank, and we need to, retrospectively assign individuals to their own family group. And this is the basis of parentage assignment. So briefly, parentage assignment is based on algorithms using genomic data to reconstruct parent-offspring relationship. And the start of parentage assignment is dated back to at least 30 years ago with the advent of microsatellite markers. As we have already seen from Costas at the beginning, microsatellite markers were among the first markers used, the most important markers used. And as you can see from the references I reported here, it was parentage assignment and using microsatellite was something that raised the attention, and it was really important for aquaculture. It was a great step for aquaculture. The technological progress made available new markers, new molecular markers, and nowadays we use only SNPs, so single nucleotide polymorphisms, as you remember from the first presentation, that have good characteristics, low mutation rates, low genotyping error rate, high prevalence in the genome. So this means that they have many advantages that make them really suitable for our specific needs. So now we have a little bit of background of what happens for parentage assignments. So let's try to analyze the process to obtain pedigree records. Here I would like to give you a kind of a step-by-step procedure, a kind of let's say a cookbook for parentage assignment. It is true that every aquaculture farm has different needs, so just follow the points as general suggestions. It represents what in general is done to perform parentage assignment. So let's start with the first point. So I mean, once we have our batch, we mix the families. So we have different individuals in the same tanks, but they belong to several families. So one tank contains individuals belonging to several families. What we can do to trace back pedigrees. We have to take a fin sample from both parents and offspring. So we just take a little piece of fin and we preserve the fin in absolute ethanol. At the same time, we can tag the animals. So obviously, we need to wait until the appropriate size of the animals. But this is important because it improves the traceability of the animals and also facilitates further data collection and phenotyping. The tag must be linked to the tube ID containing the tissue sample, and must be also linked to the data we will further collect. So every time we record something, we record a phenotype, or we do something, we perform tissue sampling, we have to recall the number of the tag of the animal. So for example, here you can see a P tag, nano tags, and the tag reader on the right. I guess some of you are familiar with these tools, but anyway, let's just have a look at them. And on the central part of the slide, you can see the tool we use to tag the animals. Then from the tissue sample, we extract the DNA. This is something that is done by specialized labs using commercial kits. It is a very technical part, and I don't spend too much time on this. It is just part of the process, just to make you aware of all the steps to arrive to a pedigree record. What is really important is that from the extracted DNA, specialized labs can also perform individual genotyping using SNP panels. In this case, we are talking about parentage assignment, so we don't need high-density marker panels. We can use panels composed by limited number of markers. We are talking about from 100 to 450 markers. And the prices are relatively affordable. We are talking about maximum 50 euros per sample. Nowadays, validated SNP panels are commercially available for several aquaculture species. The most important fish species, as you can see here, we have validated SNP panels for Atlantic salmon, for rainbow trout, and European sea bass and sea bream, but also for shellfish, prawns, and shrimps that, as correctly Costas remind at the beginning, also prawns and shrimps are really important for aquaculture. So now we are at the technical part. I mean, the most maybe difficult part. So once we have our genotyping data, we used SNP panels to genotype our individuals, and we have our genotyping data. What we can do? So we apply specific algorithms to perform parentage assignment. There are two main methods. The first is an exclusion-based method. What does it mean? When candidate parent genotypes of specific offspring violate the Mendel's laws of segregation of alleles, we exclude those parent-offspring combination. So just to remind you, from the Mendel's laws of segregation of alleles, when an organism makes gametes, each gamete receives just one gene copy or allele, which is selected randomly. But make it simple. Looking at the example here in the second part of the slide, each parent has two Lego pieces, has two colored pieces that are the two alleles, and gives one piece to the offspring. So the offspring at the end has an orange piece and a blue piece. When we then assign parentage, we can easily exclude all the candidate parents that don't have any blue or orange pieces. And this is how exclusion-based methods work. Exclusion-based methods are at the basis of several softwares, like those reported here. They are relatively simple methods, and results are easily interpreted, but they are sensitive to genotyping errors and require more loci, as only genotypes with Mendelian inconsistencies are used. So we have to just think about the pros and the cons of these methods. Going to the second method, so the second algorithms to reconstruct parentage of the individuals. The second method is a likelihood-based method. So in this case, the most likely couple of parents is chosen, and the decision is based on allelic frequencies as a ratio between probabilities. So as you can see here, the likelihood ratio is a ratio between the probability of the offspring genotype given the genotypes of the candidate parents and the probability of observing the genotype in the population by chance. So, I mean, randomly. Some softwares that use these methods are those reported here. One is really recent from 2019, APIS. The advantages of this method, of the likelihood-based methods, are represented by the higher power and the higher assignment rate compared to the first method, for example. Also with low power marker sets, but sometimes we can get complicated or inconsistent results. So sometimes maybe the interpretation could be complicated, and we can have maybe problems to have a good interpretation. We perform all the steps, and we got our, our parentage assignment, our pedigree records. What you can do now? What are the most important steps now? The most important thing that we can do once we have pedigree data is to assess the genetic health of our batch. So what a farmer could be interested in is the assessment of the genetic health of the batch of individuals. There are several parameters to assess the genetic health of a population, of a batch. We can, for example, measure the observed and the expected heterozygosity that tell us how much variation exists in the population and how that variation is distributed across the alleles we are examining. Just as a reminder, if the alleles of a diploid organism are different, it means that the organism is heterozygous at that locus, just to remind you the term of heterozygous and heterozygosity. So the observed heterozygosity is the frequency of heterozygote individuals at a locus, and expected heterozygosity is the frequency of heterozygote individuals at a locus in a randomly mating population. What is really important here is not the significance of these two terms, but is that if observed heterozygosity is lower than the expected heterozygosity, we can hypothesize the presence of inbreeding in our batch of animals. So we have already seen that inbreeding is something that we have to take care about. So we can also measure inbreeding coefficient directly using our pedigree data. So in this case, we can measure the amount of matings between related individuals in a population, and we can have an idea of how much our individuals are mated, if they are full or half siblings, or they are cousins, or so on. We can also have a measure. We can get a measure of the genetic relatedness. Again, we can have an idea of the relationship, the genetic relationship between individuals, and we can measure the effective population size that we have seen from Mark, that the effective population size is something that we are really interested in. So the effective population size is the number of individuals in an idealized population with random mating and no selection that would lead at the same rate of inbreeding as the real population observed in the real population. So the effective population size can be much less than the actual population size. And this is related to the unequal genetic contribution of individuals to the next generation. So again, it gives us an idea of the genetic contribution of our parents to the offspring. So just have a look at the first 4 points. What are the keynotes? Having pedigree data is really important to control inbreeding, to avoid genetic loss, and then to avoid the decrease of genetic diversity, and obviously to avoid the accumulation of deleterious genetic variation. Finally, the 5th point is related to the family structure. So once we have our pedigree data, we can have an idea of the family structure of our batch. So we can estimate the number of offspring for each parent, the number of families, and the size of each family. Practically, as you can see here, these are parentage assignments from PerformFISH data from CBRIM. As you can see here, from parentage assignment, we can get a list similar to this on the left, with the offspring ID and the assigned dam and the assigned sire. And then we can also derive the family ID. From this list, we can produce this big and nice table. Here we have the dams, and here we have the sires. And each cell represents the crossing between the two. And this is the number of offspring produced during each mating event. So for example, let's have a look at the orange cell. This is the crossing between dam 1 and sire 1, and we didn't get any offspring. If we have a look at the crossing between dam 4 and sire 1, here we can see that we have 40 offspring produced by this dam and this sire. This is the total number of offspring produced by each sire here, the total S, and the total number of the total number of offspring produced by each dam here, the total D, and obviously the total number of animals in our data set. This is another way to visualize the number of offspring in each family, for example, and the number of offspring produced by each parent. So, for example, in the first bar plot, we have the family ID on the x-axis, and the number of offspring for each family on the y-axis. And in the second bar plot, we have the parent ID on the x-axis, dams in red and the sires in blue, and the number of offspring for each parent on the y-axis. So, for example, the number of contribution to offspring is reported here. So, if we add a further step and we add phenotypic data to pedigree data, for example, we can measure our fish, we can have an idea of the body length, we can have an idea of the body weight, or we can have phenotypic data that require the slaughter of the animals, so fillet yield or disease resistance. Here, this is an example of a challenge test for disease resistance. If we have also the phenotypic records along with pedigree data, we can estimate genetic parameters of the trait of concern. So we can estimate additive genetic variance, phenotypic variance, and then the heritability that we have already seen that it is a very important parameter for breeders and for to organize breeding programs. And we can also have an idea of the genetic and phenotypic correlation between traits. We can estimate breeding values, that is the prediction of the genetic merit of an animal for a particular trait relative to the other animals in the same population. We can estimate the differences in the response of genotypes to different environments. So the G by E interaction, the genotype by environment interaction. And we can then think about implementing breeding programs and also perform optimal contribution selection. So this means that we can maximize genetic gain in the next generation, but having a look at the rate of inbreeding. So we constrain the rate of inbreeding, trying to maximize the genetic gain. So what this means is that we can restrict the relatedness among offspring in the next generation. And finally, what we can do is implement genomic selection. That is the second part of the presentation, and as I told you at the beginning, the two topics, so parentage assignment and genomic selection, are related to each other. So let's go straight to genomic selection. And again, just a little bit of background, just to understand a little bit more. Traditional genetic improvement programs rely on phenotype records and pedigree records to predict breeding values through lab methods. The problem here is, what about traits that requires the slaughter of the animals or challenges? So if you want to measure carcass traits, fillet yield, or we want to assess the disease resistance of a population of the animals, and we have to perform a challenge test. As Mark told us, if we fillet a fish, this fish cannot be a good breeder since it is dead. So in this case, we talk about blood procedure with seed selection. So in this case, we have only the phenotypic information of full or half sibs of breeding candidates. So the selection of individuals is based on family EBV, family breeding value. This means that each family member has identical EBV with no distinction across members and no possibility to rank individuals within families. So this family selection method is currently the most used approach, but it is not very efficient or precise. It is not very efficient because results in a quite low accuracy in breeding value estimation. We can exploit a limited amount of the total genetic variation, and we have a low selective response. Furthermore, the susceptibility of this method to further to different components affects the efficiency affect more the efficiency, like environmental variation or environmental variation that overcome genetic differences across families, or the family size. It's something that can affect the efficiency of the methods, and also the necessity of a satisfactory number of families to keep inbreeding rates at low levels. We have already seen that the number of animals we need is much more than what we expected. So it is something that we really have to keep in mind. During the last 10 years, the technological progress have provided marker maps for different aquaculture species. And in this case, we are talking about high density marker maps. In other words, SNP arrays covering from 1,000 to million SNPs spread in the genome of the animal. So we are not talking about few hundreds of SNPs as we have seen, we have already seen for parentage assignment, but we are talking about a high density marker maps. The availability of these new high density marker maps makes individual genotyping feasible at reasonable cost. I mean, it's not as cheaper as the SNP marker arrays with limited number of markers, but I mean, the technological progress is running, so it is possible that we can have individual genotyping at really reasonable cost. This availability of high density marker maps allows the advancement of genetic approaches, providing alternatives to the traditional methods we were talking about. So we can use marker-assisted selection and genomic selection. So we have new methods in selective breeding that can advance the selective breeding program. Genomic selection means that the selection of the animals for breeding is based on EBV calculated from the joint effects of genetic markers covering the whole genome. Genomic selection estimates the effect of a large number of chromosomal regions defined by a dense panel of genetic markers spread in the genome without paying attention to the statistical significance of the effect. So it is something that is really important for polygenic traits, for example, and aggregate such effects in the prediction of the genetic merit of breeding candidates. Also, it considers all markers as potentially linked to at least one quantitative trait loci affecting the trait. So it is something that is really important for selective breeding. So here I just want to give you some terminology. So just have we want to see just the terminology here. So we produce the batch of fish. This batch is randomly split into two groups of full or half sieves. So the reference population and the breeding candidates. And among breeding candidates, we will select the new breeders through genomic selection, and we will see how. So we can update and integrate the cookbook I have already shown you, adding the steps related to genomic selection. So the first steps are the same. So we perform a tissue sampling, we tag the animals, we extract the DNA from tissue samples. We perform individual genotyping. In this case, we are using high-density SNP arrays and not just some hundred of markers as in parentage assignment. We assign parentage, and then we phenotype the animal. We record the phenotypes of the animals. The first 5 points, so from the tissue sampling to the parentage assignment, are performed on reference population and breeding candidates, while the phenotyping is performed only on the reference population. From step 7 to 9, so 7, 8, and 9, these steps explain genomic selection. So we estimate SNP effect using reference population genotypes and phenotypes. We derive a prediction equation, and then we estimate the genomic estimated breeding value of breeding candidates. And this is how we select the new breeders. But we will see all these steps in detail. So we split the animals of our batch in reference population and breeding candidates. So these two groups are full and half sieves. On the reference population, we perform tissue sampling. So we take a piece of film, we tag the animals, we genotype the animals using high-density marker arrays. We assign parentage, and we record the phenotype. So let's say that our trait of interest is, for example, fillet yield. So we measure the fillet yield. On breeding candidates that are maintained safe in the farm, we perform the tissue sampling, we tag the animals, we genotype the animals using, again, high-density marker arrays, and we assign parentage. So the reference population has phenotypes for traits of interest, so has phenotypes for fillet yield, for example, and genotype. Phenotypes and genotypes are used in regression models to estimate SNP effects, and a prediction equation is then derived. It is a prediction equation that uses markers as inputs and predicts breeding value for fillet yield. So breeding candidates have genotypes. I remind you, breeding candidates have genotypes. The prediction equation we derived is used to calculate their GBV, genomic estimated breeding value, in order to select the new breeder with the best performances in terms of fillet yield. So this is more or less what, how genomic selection works. So in terms of main advantages of genomic selection, selective breeding based on genomic information is suitable for polygenic traits. So it overcomes problems related to the polygenic architecture of the traits, as it estimates the effects of a large number of chromosomal regions without paying attention to the statistical significance of the effect. But as I told you, trying to aggregate such effects in the prediction of the genetic merit of a breeding candidate. Genomic selection overcomes problems related to the unfeasibility of breeding candidates phenotyping. So it is suitable for all the traits that cannot be measured on breeding candidates. So all the traits that require the slaughter of the animals or a challenge. Phenotyping needs to be performed only once for the individual of the reference population. It guarantees the assessment of the individual genetic merit for the trait of concern, and the within family components of the genetic variance can be exploited. In this way, the selection of breeding candidates can be greatly anticipated in life, and this is very useful when we run a breeding program. So just to conclude, to have our take-home messages, parentage assignment is important to control inbreeding, to avoid genetic loss, and then to avoid the decrease of genetic diversity and the accumulation of deleterious genetic variation. It is useful also to estimate genetic parameters of the trait of concern and to maximize genetic gain in the next generation, constraining the rate of inbreeding through optimal contribution selection. Genomic selection is suitable for polygenic traits, is suitable for traits requiring slaughtering. It allows the exploitation of the individual genetic merit for the trait of concern, and allows to anticipate the selection of breeding candidates. So both parentage assignment and genomic selection are really important for those who are interested in organizing a breeding program in aquaculture. So I think I'm done. I will be glad to answer to any question you may have.
Thank you very much, Sarah. Very nice talk. Thank you again. I don't see a question related directly to your presentation.
They will come.
They will come. I don't know if you can reply to a question from Nanteza. I don't understand well about what brings unbalanced families.
What brings unbalanced families? I mean, in terms of contribution from the parents, I think if I understood well the question. I think unbalanced families could come from a lot of reasons, actually. As Mark said before, when we are in a situation of breeding, when we want that a parent produce eggs or sperm, it is not always what we want. I mean, the individual can produce, but it can also not produce anything. So this is something that we can have in mind. If we are crossing individuals, the imbalance in the offspring could be could arrive from from our crossing. I mean, from wrong crossing between individuals. So if we cross just few individuals, few parents, it's something that can affect the contribution. So
exactly, and at the end of time, the smaller, let's say, participation
yes.
of broodstock in the next generations, little by little, caused by
Yes.
these unbalanced families. Joaquin has also a question for you. If there is any relation between GBVs and EBVs, or they are just used in different cases?
Yes, there is a relation between the two. Obviously, there is. It is something that we use, for example, to assess the accuracy of our prediction equation. When we have our prediction equation and we have our GBV, we can correlate GBV with EBV to assess how well our prediction equation is working. So if we have a good accuracy, it means that our prediction equation is working well. I don't know if I answered the question properly, but we cannot hear you, Kostas.
Thank you, Sarah. We'll go to the questions that have been posted to the Q&A window. There is Joaquin again, who asks if there is an approach to cost efficiently study the metabiomes. For this question, Joaquin, there is usually a microbiome analysis in which we prepare some special, let's say, amplicon libraries for 16S or 18S or other known markers for the bacteria, the microbes under question, and we more or less screen the content of the gut or the content of the tank. So we filter the water out, and using what is remaining, we are studying what's inside. So there is kind of bulk analysis, and at the end we have a rough description to the level of the genus, and for few species we can identify exactly which are the microbes or the content in the gut and so on. There is another question that was asked by Greg. Hi, Greg. What are the current drawbacks still with imputing SNPs using low coverage whole genome sequencing for genomic selection? I don't consider myself a specialist for imputation or genomic prediction. Maybe other colleagues can reply to you. But we know that imputation can infer unobserved genotypes in a sample of individuals that have higher genotypic density from a SNP array or low coverage or the normal whole genome sequencing. But they don't provide the understanding of the causal mutation that can be obtained by annotation of the highly significant sequence variants. So there are two options. One is to impute the SNP array genotypes to sequence resolution based on a reference population of a small number of deeply sequenced relatives. And the other option is to impute from this big number of sparsely low covered sequenced individuals that we have from the low coverage whole genome sequencing, and compared to SNP chip data, this methodology, the low coverage whole genome sequencing, is able theoretically to expose the segregating sequence variants and mitigate the certain bias for these SNPs compared to the SNP array. And because SNP arrays do not contain all the genomic variants, but a few hundred thousands of them, they may include, but they may not include the causal mutations. But again, as it is a reduced representation library, we may still miss some of the variants. We still have to impute, I guess. I don't know if Dimitris, Sarah, Mark have a reply on that. Mark, you raised your hand.
Yes. Well, I think it can be very useful to impute a whole sequence when you are looking for to refine the position of a QTL to find the causal mutation. If you have low coverage sequence on all the parents and then SNP genotype or SNP chip genotypes on the offspring which have the phenotypes, that can be very useful. But it's really for this discovery, because if you are looking at genomic selection, the usual resolution of the SNP chips is enough to have a good coverage of the genome and have so for practical selection, I think you don't need this imputation, or the option to use imputation for practical selection is in fact to use the medium density, a few tens of thousands of SNPs on all the parents, and then a low density like 1,000, 2,000, 5,000 on the offspring, and then impute the medium density on the offspring. And then this can significantly improve the precision of genomic selection. But I think that going to the sequence for the practical aspect of genomic selection is probably not useful.
Thank you, Mark. There's a question probably for you from Ezra Biakora. In the tilapia spawning cycle, it's most definite that breeders won't spawn at the same time. What's the effect of this on the selection growth performance trait, considering the size, weight of fingerlings or juveniles in a breeding program?
Basically, there are two solutions to this issue. It's true that in tilapia you cannot have a synchronized reproduction. So there are two ways to deal with that. One way is to do only within family selection. And in fact, you don't select across the families because you consider that the genetic value between families is too much influenced by environment, time, etc. And the other solution, still you need to have the family information, is to use this family information to make a kind of regression on the spawning date to correct the values that you have for each individual by the average value of the same family spawning on the same date. And in this way, try to normalize the growth of all fish. But of course, you cannot just select the fish or based on their own simple body size, because if you do that, you only select the families which are the oldest.
Thank you, Mark. There is another question from Joaquin. Overpressure for commercial broodstock could be a concern if you have nucleus population with higher diversity. It's a question. Overpressure for commercial broodstock can be a concern if you have a big.
It depends what you call overpressure. Overpressure in breeding nucleus is always a concern, because if you make very high selection pressure, then you generate very disequilibrated families. You increase risks also, because, you know, practical things, there is always a risk in a fish farm to have traceability problems and to have some fish from another batch coming into some batch. And if you do overpressure, you have quite a bit of chance to select those fish that come from somewhere else. So in a nucleus, it's always a problem. Then in a commercial broodstock, if you talk about the multiplication step, this is probably not that much of a problem, and it can be even useful to have a very strong pressure to increase the genetic level of, but only the fish that are used to produce the juveniles, and not the ones that are used after in the nucleus. Of course, the more you make pressure, the more you are at risk to make a mistake. But if it's a mistake that is just for one batch of production fish, it's not a huge issue. Whereas if it's for renewing your nucleus, it is a huge issue.
Thank you, Mark. Concerning your presentation, it's a question from an anonymous attendee and Sachi. The link between lower fat content in adult fish, which means higher water content, in fact, and feed deficiency, if there are publications.
There is not yet a publication. In fact, there is I would say, a concept publication by Antti Kauser from Finland and Peter Knapp from the Netherlands, which explains that in general, fish with or individuals with more fat are less efficient. And concerning the results that we got in PerformFISH, there is a publication which is now under revision, and well, I hope that will be accepted soon. So yes, there will be something.
Okay. I don't get the question from Sachi. Compare gift tilapia and Atlantic salmon. Maybe it was for the previous question, if there are studies. There are tilapia studies for the fat content? No. No, it's only for salmon, you said.
Yeah.
From an anonymous attendee, I read the phrase, nowadays robust breeding is the way to go, avoiding extreme selection for a single trait, even if possible, to allow aquaculture in various environments, RAS, intensive, extensive, under different pathogen environmental conditions. As such, loss of genome-wide genetic variation should be measured, controlled even more to allow long-term breeding goals. How is this implemented nowadays, or what's the best strategy?
I have two replies for this.
Yeah. I
One,
don't know, you, Sarah, who also...
No, but I think there is one, which is increase effective population size, because that is the basis. I mean, all the rest is things you can do to arrange a little bit, but the main driver is always this one. And the other, I think Sarah could answer, is about optimal contribution selection.
Yes, I'm not really sure that I understood well the question. How
is the genome-wide genetic variation should be measured, controlled even more to allow long-term breeding goals?
Okay, I think that yes, optimal contribution selection could be a way. I mean, we can maximize the contribution to the next generation, but we can constrain the rate of inbreeding. So this is a way to avoid genetic loss. So I think this could answer the question, I think.
Yeah, but one thing that is really needed to understand is that, okay, you can constrain the rate of inbreeding to a certain level, but if your population is too small, it will not work anyway. I mean,
anyway, yeah, for sure. Yes.
Sarah, yes, this question is for you. If the reference population can be production fish, provided that the phenotype is collected at the same time as the fin clip is collected.
Yes, I think that the reference population could be production. I mean, we have to understand what is production, because, for example, it is not an example from aquaculture, it is from swine production. But if we have to measure the loss during salting procedures, these are animals that comes from a reference. This is the reference population, but they are going to the production. So we can do the same for fish, but I mean, when we sell fish, often we sell the entire fish. So if we have to measure the fillet yield, I don't think that this could be production fish. They just need to be experimental fish. So it really depends upon the trait we want to measure.
And Francesco also thanks everybody for the nice talks. And he asks if genomic selection works for polygenic traits. But what about the trait that is not at the individual level, but that is measured at the population level, such as overall % survival or % of females?
Okay, the first question is really simple. Yes, genomic selection works properly for polygenic traits. The second part of the question, what I know is that we always need individual phenotyping, but I don't know if Mark can help me.
Yes, well, in fact, the two traits that Francesco is mentioning are different. % females is not a trait. What is the trait is sex. So is an individual male or female? And this trait, sex, can be perfectly managed with genomic selection. It's the same as disease resistance, dead or alive or male or female. It's the same type of trait. For this, there is no problem. General survival is more difficult because in general survival, you generally don't have the whole of the trait because you only know which fish have survived. So especially if you think of larval survival, you know nothing about the genotype of the fish that did not survive. And you also don't know what was the composition of the population before mortality happened. So it's very difficult to measure. But if you talk about general survival after the fish have been tagged, for example, here you have the phenotype. If you have tagged the fish, you have some fish that in the end are survived and some which are dead. And this is a trait that you can analyze also with genomic selection.
Thank you, Mark. An attendee, Thila Karatne, asks if there are common modern genomic processes or activities specially in use for sea bass farming. I mean, everybody can ask, can reply to this question. Yes, there are. The European sea bass, because that is the fish species that we're talking about, it's not the Asian sea bass, has been targets for genomic approaches in the last 15 years, 20 years. And little by little, after constructing the linkage maps, we are now at a very good level of the genome assembly, and dozens of studies that have analyzed growth, FCR, disease resistance, and lots of studies in heritabilities. I don't know, Mark, if you can add something on that.
I can add something practical. I mean, that genomic selection is being implemented in some breeding programs. The two sea bass breeders that I know in France are using genomic selection now for at least 3 years. Yeah. And I think it's starting to be the same also in other countries.
Thank you. There are still 3 questions, but every time we answer to one, another one pops up, and I'm afraid that especially you will lose your lunch break. I don't know. Are we going to continue? For how long are we going to continue, or we're going to take over these questions later? Let's reply to another one. How can co-selection of negative deleterious traits be controlled efficiently when target trait selection is pushed as far as possible? In cattle dogs with associated diseases due to extreme selection. Like in the case of cattle and dogs.
Well, I think we can gain from the experience, and chicken also, and poultry, they have had a lot of problems. We can gain experience of what has been done before in land animals. And we know that for some traits that we select, there may be issues. For example, if you select for very high growth rate, there is usually a negative trade-off between growth and reproduction. So you expect that at some point the reproductive performance of the fish will decrease. And so if you are doing very strong selection on growth rate, then you have to monitor at least what is happening to see if some deviation starts to appear. And if you see that some deviation appears, then you can integrate this trait in the breeding program to balance the selection and avoid to have your population deriving to something that is not viable. Similar to for fillet yield, for example, we know that in some cases selecting for higher fillet yield will lead to fish with a smaller head. At some point, head cannot reduce to nothing, especially because you have the respiratory organs in the head. So when we do selection on fillet yield, there is always also a monitoring of head size to be sure that where we are gaining fillet is not at the detriment of head size. So it's possible to manage this.
From Federico, there is a simple question for sea bass and sea bream, if we can improve malformations. We know that it works for growth and disease resistance, and if we can improve malformations, to my knowledge, this is a criterion of selection in many breeding companies, malformations.
But yeah, Malformations is not very easy because, in fact, you know, we still don't know what causes all malformations. We know some things that cause malformation, and there is no general solution to malformation in sea bass and sea bream farming today. And in fact, you have several factors that can cause the malformation, and it is very likely that the genetic sensitivity of fish to malformation, which exists, is not the same depending on which is the causing factor of the malformation. So it is very difficult to have fish that would not be sensitive to malformation, whatever happens to them. And what we have seen in previous research is that there was quite some genotype by environment interaction with malformations. So which means that, okay, selecting for malformation in one condition, one environment will not necessarily give large gains in another environment. So it's not easy.
Yeah, not to mention that sometimes malformations at a certain age or during grow out can be overcome at later stages.
Yes, yeah, they can change over time, worsen or heal.
Yeah, and also diet could affect, or could be a cause of malformation. So yeah, there are a lot of factors.
Thank you, Sarah. And the last one from Greg. How can the effective population size be increased safely, efficiently in a suboptimally started breeding program, for instance, too low NE to high intensity without losing too much acquired genetic gain? What do you think, Sarah? In fact, if we can, after a while, and reaching a good point of improvement, if we realize that we have a low NE, how we can increase it safely, efficiently?
I think it's it's a bit complicated here. I mean, if we start with a suboptimal number of animals, I mean, it's not really easy. It's
yeah.
I don't know. It's quite complicated. Yeah, we.
What do you think, Mark? Before getting to lunch.
Again, there are two answers to that question. The first one is that what is the most important in management of inbreeding and of loss of genetic variation is what adds on from generation to generation. So if you have your breeding program and you see that you have not done well, but you don't have problems yet, what you can is increase the size of your population at each generation to avoid losing more. And if the situation in terms of phenotypes is satisfactory, then this should work more or less. If you start to be into problems, there is no other solution than to import genetic material from elsewhere. So preferably, of course, from another breeding program. But then you have to find a way to do it properly, in agreement with the other breeding program or not in agreement. It depends. I mean, but yes, you have to import new genes, new material, and it's better if it's from another breeding program with objectives which are more or less the same as yours, because you will not lose too much. But at some point, if you have something very specific, you lose.
Thank you, Mark. And let's take the last one. No,
there is no last one.
No, okay, but that's the last one for Sarah. If there is a cost-effective analysis with regards to accuracy and genotyping methods.
Well, yes. Nowadays we are the technological progress is running. So we can have really genotyping data at I won't say cheap costs, but I mean they are affordable now. And if we see, if you have a look at other species, like for example terrestrial species, now genotyping is really affordable for those species. So I think that also for aquaculture species, there will be like a good cost in the future. So in these terms, I think that if we will have more genotyping data, we will also have more accuracy in genomic selection, and also we can apply genomic selection. So
yes, I agree, and also it becomes a trend to have low density SNP panels.
Yes.
To go for a cost-effective genotyping at a price lower than that of microsatellites, let's say. So
yes.
Thank you all. So let's have a break and see you at 1 hour 15 Central European time. Sorry to have lost for the panelists a quarter of the hour to make your break. So see you in a while. Ciao.
Is it okay?
Excellent, thank you.
Yeah, okay.
Mm-hmm.
So I can start now, yeah.
Yeah.
Okay, so this afternoon we will start. This morning it was more about the principles of selective breeding and modern genomic techniques that we are using now in aquaculture. And this afternoon we will talk more about traits. And as an introduction, I will make a presentation on the genetics of efficiency traits and how we can improve this in aquaculture. So I always remember that my teachers told me that repetition is the basis of teaching. So you have already seen that this morning, but maybe not everyone was there this morning. So I will recall briefly what we do when we do selective breeding of fish. So we have parent fish that we reproduce. From that, we generate selection candidates, individuals that are evaluated for the phenotype, the traits of interest. And we will select the future parents of the next generation from those best candidates. We do that. We can do that with the phenotype. We can be helped by the pedigree, to do that, to have some more precise evaluation. And we can also have challenge fish, for which we measure something that we cannot measure directly on the candidates, like disease resistance, for example, which we also pedigree. And with this genetic information, we can assign to the candidates a breeding value for a lethal trait. That's the general principle of fish breeding programs. And so you need to have phenotypes. You need to be able to measure the trait that you want to improve accurately enough. The number of candidates is typically high. It counts in thousands. Here I put 3,000, but sometimes it's more like 10, 15,000. So you need the phenotype to be efficient and sufficiently low cost. It's a long-term process. And of course, the pedigree improves our capacity to do breeding, but has a cost. Okay. If we look at the early fish breeding program, the first modern fish breeding program that has existed is the one started in Norway in 1972, which used the pedigree information. And at that time, the only way to have the pedigree information was separate rearing of the different families. So they have constructed huge holes with hundreds of tanks. And in each tank, you have one family like this. As early as 1972, they could have access to this family information, which allows to improve the quality of the breeding values. In other situations, for example, in France, we used breeding programs without pedigree, like the Prosper breeding program, which was designed by Bernard Chevassus in INRAE in 1986, and which used more the individual phenotype of the fish, so their individual growth rate as the selection trait. So less precise, but less costly. And at these times, there was one simple major goal, which was faster growth. And indeed, it worked very well because you see, this is, already showed it this morning, but for those who are not here, this is the gain in growth that was obtained from these early breeding programs. So we have some on trout, we have some on salmon, we have some on tilapia. And if I put a reference starting point of 100 for each species at the beginning of the breeding program, you see that in a reasonable amount of years, it's possible to double the growth rate of any fish species. So, okay, it works well, it improves growth very well. If we look at the traits selected today in European aquaculture, this is the result of a survey that was done during the Fish Boost European project, and which was published in 2016. The main species investigated were salmon, trout, sea bass, sea bream, turbot, and carp. And what we see here is that growth is part of the selection goal in all breeding programs, basically. And in other, and the different breeding programs add some other traits, like morphology, like disease resistance, processing yields, product quality, or maturity or fecundity. But in all breeding programs, the growth is present. But then the question is, okay, selecting faster growing fish works well. And it's true that if you go to see a fish farmer who wants to start a breeding program and ask him which trait you want to improve, it will tell you, oh yeah, I want faster growing fish. But is it really that we need to improve? I'm not really convinced about that. If you look at this picture, it's representing what you have to do the morning to go to work. Okay, you think that you don't go to work fast enough. Selection for growth is saying that the solution is this one. You change for a faster car and you will be faster at your office. In this case, it's quite evident that it is not the solution. What about growth in aquaculture? Well, how can we evaluate the economic impact of genetically faster growth? First, we have to make two important hypotheses to evaluate the impact of growth. And I really say of growth. The first hypothesis is that growth does not change feed conversion ratio. Because if you admit that growth changes feed conversion ratio, which may be true or not, you don't want to see the impact of conversion ratio. You want to see the impact of growth. So if growth does not change FCR, if I have a slow grower and a fast grower, what will happen is that if my fish grows twice as fast, it will consume 2 times the amount of feed of the slow grower. It will consume 2 times the amount of oxygen. It will produce 2 times the amount of weight. It will just do the same, but faster. The second important hypothesis, if you want to see the economic impact of genetically faster growth, is to consider that you are working in an optimized breeding program, which simply means that you cannot just stock more fish. If stocking more fish is a solution to earn more money, it means you are not optimized. Okay, you just need to stock more fish and you will earn more money. If you cannot stock more fish, it means that you have really optimized your system and you take advantage of all that is possible in your system. Then, if growth does not change FCR and if the rearing system is optimized, the key question becomes which factors do limit production? And here you can see different solutions. One very frequent limitation in aquaculture is the license. In most countries, you cannot have an aquaculture operation without a license. And in many countries, the license is a fixed annual production. You are allowed to produce 1,000 tons in this site. What happens with faster growing fish if I have a license of a fixed 1,000 tons? I have no gains, because if I want to output 1,000 tons of fish, whatever the growth rate of the fish, I will have to use a certain amount of feed, a certain amount of oxygen. I will produce a certain amount of waste, but the cost, main cost, which is the feed, will be the same if FCR is not changed, no matter if I produce my 1,000 tons quickly or slowly. If the license is maximal instant biomass, it's like this, for example, in Norway, where you cannot have more than 1,000 tons on the site at a given time, then gains are possible, because if you have faster growing fish, your system is rolling more efficiently, and you can stay with the staying at the same level of maximal biomass. You can have more output, and if output is profitable, then you get more money, then it's fine. In this condition, selection for growth is interesting in terms of economy. But very often you have also physical limitations to your production. For example, it can be oxygen limitation. In a trout farm, you have a certain number, a certain volume of water, a certain quantity of oxygen flowing through the farm, and you need to keep the oxygen to a certain level for the fish to be okay. So if this is a limitation, if you have a faster growing fish, you feed it more, it uses more oxygen, you have to stock at lower density. And in the end, you will have no gain with faster growing fish. It's the same in a RAS system with treatment of nitrogen. If you have a faster growing fish, it will produce more waste per unit of fish. So you need to have a lower stocking density, and you will have no gain or almost no gain with faster growth. So in general, we can say that growth in itself, not taking into account possible changes in FCR, brings very little economic gains. You have all a series of articles by Mathieu Besson, who studied this quite deeply some years ago. You can read that if you are interested. So if growth is not the solution, we propose a different way of
You're out again, Mark.
Yes, I'm back. Can you hear me?
Yes, yes.
Do you still see my screen or not?
No, not yet.
Okay, so I share it again. I don't know what happens, but so I said if growth is not the ultimate trait, we can change perspective and try to improve efficiency. Basically, aquaculture is to produce fish meat that we can eat. That's what we work for. So if I want to produce sea bass fillet, I will have a first important trait to care about, which is fillet yield, because to produce 1 kilo of fillet, I will need to produce 2.6 kilo of sea bass. Then a second trait of importance is disease resistance, because to have my 2.6 kilo of sea bass that I can cut and with which I can make fillet, I need to rear more sea bass because some of the fish will die during the rearing process. To do that, I will need to rear 2.9 kilo of sea bass. You may think it's not that much, but indeed, most of the time, diseases appear mostly at the young stages, and you don't lose a lot of biomass. You lose numbers of fish, but not a lot of biomass. So this figure is low, but this has some, accounts to something reasonable. And then the main cost in a fish farm is feed. So to produce my 2.9 kilo of sea bass, I will need 5.8 kilo of feed. So you see, for 1 kilo of fillet, I will need 5.8 kilo of feed to produce it. And this is a major cost. If I take the example of another species, rainbow trout, all the indexes are better in that species. And to produce 1 kilo of fillet of rainbow trout, I just need 1.9 kilo of feed. And here we are working in PerformFISH for the Mediterranean species, and I can say that my objective is to transform sea bass into a rainbow trout. In terms of efficiency, of course, not in terms of fish. One last point that is important when you talk about efficiency is also the type of feed that you feed to the fish, because we know that we have less and less fish meal and fish oil to put into the pellets that we feed to the fish. So the capacity of fish to adapt to the new types of feed that we'll have to feed them in the future is also something quite important in terms of efficiency. And then these traits maybe are not that simple to improve compared to growth. So they have both clear economical and environmental benefits, because of course, if I use 5.8 or 1.9 kilo of feed to produce the same amount of fillet, it's quite clear that my cost will not be the same, but also my environmental impact will not be the same at all. And our question is how to improve those traits with selective breeding. An important point is that some of these efficiency traits are ratio traits. If I think of fillet yield, it's the ratio of fillet weight to body weight. If I think of food conversion ratio, it's the ratio of feed intake to body weight gain. And the problem with the ratio traits is that they have bad statistical properties for selective breeding, and they are not optimal traits to use in selection. Because, for example, if I improve fillet yield, I can get the same improvement by improving fillet weight at a given body weight, or by reducing body weight at a given fillet weight. So different changes can give the same ratio. So to study these traits more efficiently, we tend to use residuals rather than ratios. If I take the example of fillet yield, on the left panel, you see the relation between fillet weight and body weight. You have a linear relationship between the two. And instead of selecting the animals with the best ratio, we will select the animals which, for a given body weight, give either a positive residual fillet weight, so which have a fillet weight which is higher than the expected mean for this given body weight, and we will eliminate those that have a negative residual fillet weight. That's what this residual trait is about. And for feed efficiency, it's the same type of thing. We can do what we can do, for example, one very frequent criterion to estimate feed efficiency is what we call residual feed intake. So what you do is you estimate the feed intake of your fish with an equation that combines body weight, because we know that larger fish in general have a lower feed intake per unit of weight, and body weight gain. And by combining these two, you can say, okay, this fish should eat this amount of feed. And you compare this with the measured feed intake. In the same way, you have a regression, and you have fish which has a positive residual feed intake. These fish eat more than they should do. So in this case, these fish are less efficient. And the ones that we want is the ones with the negative residual feed intake, so which for a given situation eat less than what they are supposed to do. You can have a symmetric thing, which is residual body weight gain, where you compare the real body weight gain and an estimated body weight gain based on the body weight, mean body weight of the fish and their feed intake. And these residual traits, they have better statistical properties, and also they are not directly linked to growth. And that is important because, as I said before, okay, selection for growth is easy. If you want to select for efficiency traits, we want to select the part of this trait which is not due to better or lower growth. So we really need this type of definition. Now, if I look for what we can do with selection for fillet yield in trout, this is an experiment that was done in a previous project, the Fish Boost project, where we did SIP selection for fillet yield in trout. In fact, it was not fillet yield, it was residual headless carcass weight. So what we measure is not the weight of the fillet, but the weight of the carcass, which is gutted and has no head, because this is very much linked to fillet, but has much less technical variability. You know that when you are filleting fish, the skill of the filleter is very important. When you are just cutting the head and removing the viscera, the skill is much less important. So you have better precision with this type of trait. So we did SIP selection for this residual headless carcass weight, and we tried also indirect selection with ultrasound tomography. With ultrasound, we measure the depth of the abdominal cavity and the thickness of the abdominal wall, and we know that the ratio of the two is a good predictor of fillet yield in trout. So based on this, so we had candidates to selection that were genotyped. We knew their family of origin. And we had SIBs, so from the same families that were slaughtered to measure the residual headless carcass weight. And then we did selection to decrease fillet yield by using either only echography, only SIB fillet yield, or the combination of the two. We had a control where we did not select the fish, and we had selection for improved fillet yield, which similarly used either the echography or the SIBS fillet yield, or the combination of the two. And you see that in the offspring, so in the next generation, we have a gradation of fillet yield related to this selection. So this proves experimentally that it's possible to improve fillet yield by an amount which is generally comprised between 0.5 and 1% per generation, depending on the species. We can see on the center panel that this was done quite a lot by reducing viscera yield, and that head yield. We talked about head yield this morning, and we don't want to decrease too much the size of the head of the fish. Head yield was not that much changed when selecting for fillet yield. So we saw that both the indirect method with echography or the method using SIB selection were efficient. So we can select for fillet yield in trout. In PerformFISH, we did not do this type of experiment, but we estimated the heritability of residual fillet weight or residual headless carcass weight in sea bream on the breeding population of Ferme Marine du Soleil, which is the new name of Ferme Marine du Douai that many of you know probably, a French breeding company leading on sea bream. And so the heritabilities that we get are moderate, I would say, and this gives an opportunity to gain something comprised between 0.5 and 1.5% per generation in fillet yield. You can see I have an article in Aquaculture in 2020 which details how you get from the heritability to the genetic gain for fillet yield in fish. So yes, we can increase fillet yield by selection. So that's first efficiency trait, it's okay. The second one is selection for disease resistance. I will not get into big details for this because Stavroula this afternoon will give many more details of what was done in PerformFISH, but I will just go through a little bit of the principles and what has been done. How to select for disease resistance? You can select of survivors. So you have your fish, you expose them to the pathogen, a few of them die, and you keep the survivors as broodstock. This is not very much used because when you do this, you contaminate your broodstock, and that's really not what you want to do in a breeding program where you try to protect your nucleus as much as possible. But this has given some interesting results, for example, for selection of white spot disease resistance in shrimp or selection against IPN in salmonids. The other solution is SIB selection using family links. And if you know to which families the survivors belong, you can select the same families in your candidates to selection. And this has been shown in many species to be able to generate a significant gain in survival per generation. The last way to do it is to use QTL or genomic selection. In this case, you don't use family links, but you use evaluation of the, say, the quality of the genome of the fish. And you can identify which fish have a good quality for resistance to a disease. And then among your candidates, you genotype them and select the fish that have the same good quality. For genomic selection, we don't really have results in our species, but using QTLs, there have been very spectacular results on salmon in Norway, where the diffusion of QTL-resistant eggs provoked a drastic reduction in IPN outbreaks in this species in Norway. So we can select also for disease resistance, and you will have more details on our favorite species later in the afternoon. Now, let's go to feed efficiency. You have seen in my graph that it's really one of the major traits of efficiency, and it is not implemented in breeding programs. Why? Well, if you remember what I said several times already, you can select any trait that you can measure. So to select for food conversion ratio, we should be able to measure our fish for food conversion ratio. Food conversion ratio is a ratio of feed intake to weight gain. It's easy to measure weight gain. You tag the fish, you weigh them at one time and then at another time, and you got the weight gain. But when you distribute feed in a tank or in a cage, you have absolutely no way to know which fish is eating what, and especially over a long period of time. So we cannot measure feed intake in production conditions. So we have no individual FCR, so we cannot select for improved feed conversion ratio. Several methods have been tried to overcome this problem. One, for example, was X-ray. So you use a labeled feed that you can see with X-rays. So you feed the fish, you capture them, you X-ray them, and by counting the beads in their stomach, you know how many pellets each fish has eaten. The problem is that it's very precise, yes, but you measure only one meal. It is stressful to the fish. It needs to be repeated. And so probably it's not integrative enough to be very efficient on the long term. A colleague of ours, Hugues de Verdal from CIRAD, has developed a method using video recording of meals with tilapia. He has tilapia in an aquarium. They are tagged with color tags, which you can see on the right. And he distributes feed pellet by pellet and records that on the video, and then spends hours and hours looking at the videos and counting how many pellets each fish has eaten. It's extremely tedious and can be done only for short periods, like days. So you see, it's complicated. But maybe we have a first good solution. If growth does induce better FCR, then we would be saved. It would be fine. Just selecting faster growing fish would give better FCR. Is that the case? Well, if we look at what we have in the literature right now, we have quite little data comparing food conversion ratio of selected and unselected fish for growth, but there is a few. And we can see that in 2 salmons, we have an effect. So selection increases growth rate and decreases FCR, so that's good. But in other experiments, we have absolutely no effect. So we have a clear effect of selection for growth on weight gain, but absolutely no effect on FCR. So what this tells us is that growth may contribute to a better FCR, but is not enough. And probably not enough, it's also probably system and species dependent. So finding a way to record feed intake remains necessary. To do that, we have first tried to use indirect trait, which is weight loss as fasting. We did that in sea bass. And I would say that, okay, you don't know the individual feed intake of fish, except in one simple situation. If you don't feed the fish, you exactly know that each fish has eaten 0. And with that, you can predict that fish that lose little weight during fasting are the ones with a lesser basic metabolic rate and probably will be the ones with the best feed efficiency. And on CBAS, we confirmed this. We showed that the fish which had a low weight loss during fasting, which are represented here in orange and red, have a negative residual feed intake. So it means that they are more efficient. We also showed that this weight loss at fasting was heritable. And we did 2 generations of experimental selection, 9 years, to arrive to the conclusion that, yes, if we compare the breeding value of fish for weight loss at fasting with their FCR, when these groups with different EBVs are put in different tanks, we have a significant relationship. And the fish that lose less weight at fasting also have a better feed efficiency. But still, we would like to be more direct. And this is what we did in PerformFISH. What we did is that we used sea bream from the Fairmine du Soleil breeding program, and we reared them in individual tanks where we fed them with a known amount of feed, which was restricted relative to the normal ration for practical reasons, and also because it had been shown in other species that growth under restricted feeding is a good proxy of feed efficiency. And just to make you realize, restricted feeding can only be done in such conditions with individual fish, because if you have fish in a group and you restrict feeding of the group, some fish will be very much restricted and some fish will not be restricted. So it's not a solution to restrict feeding in groups. But if you have feed isolated, then you can have them under restricted feeding. So we put the sea bream into this aquaria. They were habituated for 1 month, and for 1 month, we had individual measurement of feed intake, body weight gain, and then FCR, with daily collection of all uneaten pellets and precise quantities of feed given every day. Out of more than 600 fish that we tested in the system, only 566 adapted to the system and were able to grow under these conditions, which are quite stressful for the fish. And from this, we got more than 500 phenotypes of feed intake, body weight gain, and FCR. We showed that we had good phenotypic variation of FCR, and we could calculate on the diagonal the heritability of the FCR, of the restricted growth, and of the residual body weight gain, which is another measure of feed efficiency. You see that all 3 are very much linked, and they have a decent heritability, so they can be selected for. One interesting point is that as the growth rate under restricted feeding is very much linked to FCR, it may be possible to do selection in the system without collecting uneaten feed, because you need to collect to know FCR. But if you just want to know growth rate, you don't need to collect. We also validated that this had an impact on the growth of fish when they were reared in normal conditions. So for all our fish, we classified them by relative feed intake in % of their body weight to relative weight gain, and did different classes. And in each class of feed intake, we selected good fish, which had good weight gain, and bad fish. And we could see that when we have these fish in groups, in the first measurement period, we had a good agreement. The good fish had lower FCR than the bad fish. Then we had some experimental problems. It was more difficult. The rankings were perturbed. And in the end, if we consider the full period, we have a small advantage to the good fish, but this is not statistically significant. Then we did more the residual body weight gain system, where we looked at the expected weight gain per fish compared to the observed weight gain. And here we have something a little bit clearer. where even over the whole period, even though the things are perturbed in the middle, we have a positive effect of selecting the good fish compared to the bad fish on this feed efficiency. We also looked at correlations with traits on commercial size fish, on our experimental fish themselves, or on sieves reared in cage in normal conditions. And we can see in both cases positive correlations with harvest weight. This is good news. And negative correlations with traits related to fat. So this means that the traits that we want to improve in the breeding program, so faster growth and lower fat, are nicely correlated, favorably correlated with feed efficiency. So this gives good prospects for the implementation of selection, even though the correlations are not very precise and are not very high. With our colleagues from Spain, we did a deeper characterization of the high feed converters versus the low feed converters, using especially an accelerometer tag that is put on the operculum of the fish, which was developed in the AquaExcel 2020 program, which showed us that the high feed converter had a higher respiratory frequency, and also at the maximum respiratory frequency, which was later in the day than that of the low feed converters. And using expression of selected genes that they know are important for the metabolic efficiency of the fish and for muscle growth, they could show also that the faster growing high feed converters had more a lean type of phenotype and were more prone to accumulate muscle. So all in all, this also gives a nice picture of what we have to do to have our good converting fish. Last point is adaptation to plant-based diet, and also in PerformFISH, we did a trial with sea bass from the Gravelines EMGI breeding program, which were fed 3 months on a special feed, which was very low in fish meal and fish oil, or 3 months on a standard feed, which was much higher in fish meal and fish oil. So the arena feed, A feed, is more representative of what could be a future feed for this species. And these fish, after these 3 months on different feeds, were challenged for Vibrio RVI at the Fortier platform in Brest in France. They were assigned to their parents with SNPs and API software, and we could see that, well, both the survival to Vibrio was heritable with both feeds, a little bit apparently higher with the new feed. And also, interestingly, that the genetic correlation between the survival with the two feeds was not very high, less than 0.8. And this shows that there is specific adaptation for Vibrio resistance with the plant-based diet. So there is an interest to select fish using a more plant-based diet than the current standard. And like this, we will have future fish which will have better Vibrio resistance when fed the future diets. We could show also, I already showed that this morning, the potential of genomic selection for efficiency traits. On our CBRIM, where we studied feed efficiency as residual body weight gain, we could see that the accuracy using genomic blob, so where the fish were genotyped on the 57K SNP chip, was higher than the accuracy using pedigree blob, so using the traditional way of doing family and SIP selection. And we could achieve a very good accuracy of 0.5 with only 500 SIPs evaluated versus usually amounts of more 1,000 in SIP selection with pedigree data. So in the end, how much can we gain in one generation? Well, if I put all this together, if I put all this together, I do a selection pressure of 25% on each trait, which makes a total selection pressure of 1.6%. We think we could gain on fillet yield, disease resistance, and feed efficiency, small amounts every time. But if you put that all together, that makes in total 5% less feed to do the same amount of fillet of sea bass in one generation. So it's not as good as what you have with growth, where you can have 10, 25% per generation, but it's pure efficiency. So in terms of economic and environmental gain, this is considerable. Don't forget that genetic gain is cumulative. But one problem is that we still don't know how all these traits are correlated. So if it's easy to select for all these traits at the same time in the same breeding index. So in conclusion, I can say that efficiency is both economically and environmentally beneficial. The major traits are feed efficiency, processing yields, disease resistance, and adaptation to alternative diets. Some progress may be expected by correlated response to selection for growth. So I spent a lot of time to tell you that selection for growth is useless, but in fact, it's not that useless. It can have some positive impact on efficiency. It will be important to study G/E interactions because we have small gains for each trait. So how will they be translated in different and often suboptimal rearing environments? And just to conclude, this is a major key for a sustainable growth of aquaculture production in a finite world. And of course, I show you a lot of results, and this is a real teamwork with people from many institutes of companies to have obtained all these results. I thank you for your attention. And this time, I'll have left no time for questions, I'm afraid.
Thank you, Mark.
No?
Yeah.
Oh, it's okay. Okay.
Thanks, Mark. Well, I will continue talking a bit of how to incorporate G/E in the genetic evaluation. I will continue a bit from the introduction that Mark did, which was very nice, and then go into definitions of G/E. For example, what you want to see what animal breeding achieved in the last 50 years, you get the most selected species broilers from the poultry industry. As Mark showed, for example, in terms of carcass gains, but we see it now in growth in live weight gain, for example, which publication 3 years earlier by the same author. It was, for example, 56 days, which was the target slaughter date in 1950s. The 1957 products, or hybrids, as we're calling it, with no selection, and compared with the 1991 product of the same company, which is Rosbreeders at the time, Saviagen today. It was 99. It was the 1991 hybrids was 3.5 times heavier, and in 2001, 4.36 times heavier. That's about 3.5 genetic gain per year. And in a very nice review by my ex-super PhD supervisor from Edinburgh, only 10% of growth in the total in the last 30 years when he wrote that review was induced by nutrition. The rest of them was just because of the selection. And similar results were in other traits also, in other species also, and also in crops. So it seems that it's a powerful selection is a very powerful tool in improving productivity, and of course other traits. And when I say one picture, 1,000 words, this is the same age chickens. The big one is a broiler. The small one is for egg purposes. And the one that was selected for high growth rate, the other one for small size to fit earlier, a few years ago in cages, and high fecundity to produce a lot of eggs. So all the difference that you see between them, it's due to selection. So and I can verify, for example, by working in Naviagen, that the gain that the Havenstein shows is sometimes even larger, and that added genetic variations is attributed to the trait, as we call it, the quantitative genetics, according to the questions that we had before, never stop, still there, still have the same heritability, still have a genetic gain every year. So it seems that selection is not minimizing our genetic diversity attributed to the traits that we selected, of course. Now going to the GBI, seeing the definitions, in general, when what we call GBI, when a breed or a variety responds differently in different environments and or different environmental changes that we impose to it, it will be more specific when two different genotypes respond differently in any kind of environmental variation. More usually and distinguishably observed in plants rather than animals for protection traits, and in plants, but we call it also phenotypic plasticity, these differences in performance in different environments. But it can be observed better in disease traits and in some nutrition-related traits in animals. It is usually depicted using a norm of reaction or reaction norm graph. And you can see some of them now. For example, you can see, for example, that is a very strong genotype by environment interaction. For example, in the line A, there is no genotype by interaction on line B, as in D, and it's negative in the A, and it's positive with E, lines E and C. For example, there's a better line B, but doesn't has any genotype by interaction as the D. Here we have a very strong interaction. Here we have not so strong interaction between the two differences between the lines. You will see it in a more classical graph where we have a strong interaction. For example, in one genotype A in the very poor environment is performing very poorly and very good in the other extremes, are in a very good environment, and vice versa. Genotype V is very good in the poor environment, but very bad in the good environment. If we see it independently, for example, there is no G by E between these genotypes when the lines are parallel. There is some G by E when they have an angle between the lines, and when they're crossing, there is a strong genotype by environment interaction. Why is more difficult to be observed in animals? In animals, in farm animals, we have more controlled farming environment. So selection of suitable breed or line, you can select a suitable breed or line to your environmental conditions. In plants, for example, that we observe it more, we don't have, if we take out greenhouses, they're exposed more to environmental conditions, and they don't have the ability to move to protect themselves from environmental conditions, so the animals they have. Does GBA has an effect on productivity? Yes, it has when you don't use the right stock. How you will choose the right stock is the next question. The answer is availability, fast when there are options. For example, there are breeding companies that are producing the right stock for your environment in poultry and pigs, less in sheep and cattle nowadays. But also you can develop it through animal breeding. It's more slow if you try to develop it, but it's tailor-made to your conditions. If we see, for example, a trait that is not associated directly to productivity, then the GBI is not so strong if we see in terms of fertility. We have high-low fertility, for example. There is some genotype by environment interaction between the high and low fertility cows. As you see, it's not completely parallel, but, and this is the science estimated breeding values, but it's not as strong. If you see it, for example, if you compare the GBI in high and low genetic merit animals, there is some environment interactions in the solid, in the content of solids in the yield. For example, you can see that they respond better in the high concentrate feed, the low genetic animals than the high genetic merit animals than the low genetic merit animals. So you get extra gain if you feed better feed, for example, in the high genetic merit animals, the high breeding value animals. And if you see an example, for example, from stream mortality in different salinity, there is also genotype by environment interaction, not very strong, but it is concerning low and normal salinity and the survival time under these conditions. When we have a very strong effect, genotype by environment effect, is again in cow milk solids yield, when you feed the animal mainly in pasture or in total mixed ration, when you mix all the components in one, the diet, all the components in one feeding regime, and you feed ad libitum. As you can see, for example, here we have the same breed of cattle, but the one with the cycle are from the New Zealand breeding program. The one in the square is from North America. And you can see, for example, that there is the New Zealand response much better in the TMR rather than the American ones. And the reason is that the New Zealanders, they have higher time. The breeding program is based mainly on feeding the cows in pastures. So when you put them in DMR, you have a higher response. They're selected for pasture. So these ones, they're selected based on TMR, so the improvement, the change is not so big. So if you take into account the system of feeding in your breeding program, you might gain more response, for example, in your selection process. There are three types of genotype by environment effects on animal or fish breeding program. When we have the GBI to inflate the variation, but it has no effect on the genetic parameter estimation. For example, there is no effect on your variance components. For example, inflates the phenotypic variations, but equally inflates the added genetic variations. So the ratios are the same, the heritability is the same. Usually this type of GBI has no influence on the genetic valuation of the animals and doesn't have effects on the ranking of the EBVs of the animals under two different environments. There's also the second case is when the genotype by environment interaction inflates variations, but has a small effect on the genetic parameter estimation. It increases unequally the added genetic variation compared to the total phenotypic, and it has medium to large effect on the genetic evaluation of animals. It produces changes in the estimated breeding values ranking. The third type is when GBI does not have effect on variation. It has a small or no effect on the genetic parameter estimations, affects only slightly the added genetic variation. We have inflation or deflation of the genetic variation attributed to the trait, and it has a small effect on the genetic evaluation of animals. Can produce small and non-significant changes on the estimated breeding value on the ranking of them. And also, this can be corrected by the genetic evaluation model if you include, for example, the environmental parameter in your genetic evaluation model that will correct the small changes in your EBV's ranking. If we want to see it on a graph, for example, if we have the two different environments, is actually, for example, in this case, the variation is much smaller, but if you put it in the same genotypes in a different environment, the inflated variations you see the curve is spread. And the same is with the genotypes. The genotypes are then distributed in all this spread. Now, 25 years ago, when I went to Aviagen as a young geneticist, it's been ages ago, almost a quarter of a century, I'm getting old. One of the problems that I had was that the competitive breeding company although it was the best product in the market, the competitive breeding company, the second biggest breeding company in the world, was more competitive in poor environments as far as the early growth is concerned. So in the first stages of the growth of the bird, when the farmer had a poor farming management, the competitive company was doing better than us. So they asked me what we'll do to change that. Together with other colleagues in the breeding company, we suggested to create a challenge environment and put sibs of the pedigree birds of the nucleus bird in that challenge environment. And the challenge environment had many parameters. For example, it wasn't only feed. It had also, for example, apart from raw specs feed diet, it had high temperature, humidity, climate inside the hatch, the shed. With the worm cleaning the litter so often, every batch coming in, was doing it every 3 or 4 batches. It was fast turnaround. For example, when we take the one batch and put the other one, we didn't, for example, fumigate from every batch. So that has effect on survival, on early growth, and robust growth. So we actually have sibs of our pedigree birds in that challenge environment, and in this way we can get, for example, we can have EBVs for the challenge environment for our pedigree birds. If we see, for example, in a multi-trait scale, the phenotypic variance in those 2 different environments, we can see that in the early stages, in the 7 days body weight, we have an inflation of the phenotypic variation from 182 to 203. But if you go to the slaughter weight, the 35 body weight, we have a huge increase of the phenotypic variation. For example, in this case, in the early growth, it was 12% inflation, and it was in the slaughter weight 190% inflation. If we see this one, the effects on the ratios, on the heritabilities, and hence in the added genetic variation, this one produced, for example, slightly higher in the stress environment heritability, and much higher heritability for the slaughter weight. In the first case, in the early body weight, it was 31% inflation, and in the slaughter weight, it was 524% inflation. That means that although we inflate, for example, the phenotypic variance, we also inflate the added genetic variation, our estimate of the added genetic variation of the trait. And we inflate it much more than the phenotypic variance when we put it under stress conditions and an alternative environment. So the suggestion is if we have, for example, plotted the breeding values that we produced from this analysis, the pedigree EBVs and the stress environment EBVs, instead of selecting, for example, according to the highest EBV and select, for example, in the stress environment, all those that are above the 0, all these ones above the y-axis, and on the right for the pedigree birds, on the right of the, or in the far corner of the x-axis, like that, for example, that's the best breeding values for the stress environment, that's for the pedigree. We're starting to select on the combined higher EBVs. This had an effect, for example, in the genetic progress. Here you can see the genetic progress from 1997 to 2001 before we applied this kind of selection. So we had 3 grams in the early body weight in the pedigree environment and 1.7 in the challenge environment. And then when we apply the selection for both, using both EBVs, we had 4.5 grams in the pedigree birds and 4 grams in the stress bird. So that means that we have 50% better genetic progress in the pedigree environment, and 135% better genetic progress in the stress environment for our birds. You will see that in the slaughter weight, that before we apply this kind of evaluation in our selection candidates, we have 46 grams genetic progress between 1997 and 2001, and 31 grams in the stress environment. And after we apply the selection, it was 70 grams in the stress in the pedigree environment and 52 in the stress environment. And that was on the slaughter weight was 52% better genetic progress in the pedigree environment and 67% better genetic progress in the stress environment. So the results of that was to have more unbiased estimated breeding values, better early and late growth in both type of environments, even better in the early growth stages where the co-competitive had the advantage. And we produce a more robust bird, with better robustness of our product, and also animals that can perform good in any kind of environment, environmental condition, in any kind of farm management practices by our clients. Let's see an example now in aquaculture. We use, for example, there in aquaculture, we don't have so much control farming. So one of the things that we altered in this case was the feed, different kinds of feeds. The one was a normal, the control diet with high fish meals. The second one was a plant protein-based diet. The protein was coming mainly from plants. We use Cibrim, 4,253 records on growth and other traits, and 6,000 or more than 6,000 pedigree records. We use two feeds, so half of the birds, almost half of the birds were fed the normal controlled diet, and the other half was fed the plant protein-based diet. Here are the performance of these birds. For example, we can see the average in both feeds. So the one body weight measurement was on July, about 500 days post-hatching, was 456 grams with 97 standard deviation. That is the length. The growth on the period from April to July, for example, was 384 grams with 74 standard deviation. And the growth from tagging to July was 636 grams with 115 standard deviation. And that's the growth between tagging and April. And also there was recording of this formation and also body weight measurement in October, which was 650 grams, approximately 120 standard deviation. We estimated genetic parameters using an animal model, and we use the feed as a fixed effect. I use an old software because I'm a dinosaur, as you can see. Here we can see, for example, the estimate of body weight in July was about 0.47, the length of the fish 0.47 heritability, and the height 0.39, the ratio height to length 0.17, and the fat percentage is measured by electric current was 0.39. Again, the same, but the same bias covariance matrix with two different traits, the disformation 0.16 and the body weight on October 0.45. If we see that, for example, now in terms of growth in two different periods, the heritability between April, heritability of growth between April and July was 0.37, and between tagging and April, 0.47. And also the length and the fat percentage, the length highly positive correlated, and the two growth periods correlated genetic correlation of 0.65. Again, the same various covariance with one different trait. For example, that was April to July 0.37 and tagging to April 0.45 heritability. So let's keep this one's these estimates in mind to see what will happen if we try to estimate them using under the two different environments. So what will happen when genetic parameters was estimated using records only from one of the two feeds? Heritability estimates were in general similar magnitude with exception of length, and the genetic parameters are underestimated with the plant feed with the exception of for fat percentage. Phenotypic variance was inflated by 15%, and the other genetic variance was deflated, but very marginally by 2%. If we see, for example, in the control field, when we estimate the genetic parameters under the control field was 0.42, though under plant feed was 0.36. So we had a deflation in the case of the plant feed diet. Also, in the weight tagging to July, 0.749, 0.42. And if we see, for example, what we had when we use the feed as a fixed effect and estimate the various components in one model, we have a slight underestimation in the case of the control field and even bigger in the case of the plant feed. And the same happens also in the opposite happens, overestimation, that's the fourth two. And tagging July also with overestimation in the control feed, underestimation in the plant feed diet. Then we analyze the traits, consider the traits as different traits into the different feeds, and we estimate the heritability, and we're actually of the same magnitude for the same traits. The trait for growth from tagging to July was 0.49 for the control feed and 0.42 for the plant feed. And the genetic correlation between those two traits was very high, was almost unity. So when feed was used in as fixed effect in the analysis. So that means that when we're measuring the same trait genetically. There wasn't any difference any difference as traits. However, when we put the feed by family interaction in the model, it had significant effect. So, for example, using the growth from tagging to July. And similar results were observed also with other traits. So, is there any genotype-environment interaction? We didn't have any large inflation of variance. The added genetic variance is also marginally deflated, and the trait is highly correlated between the feeds. So, still the question: is there any G/E effect on our genetic evaluation? On the other hand, we observed significant family by feed interaction, and we wonder if it is possible any effects to be confounded within the family effects when we do the genetic evaluations. And therefore, by this confounding effect, we have masking of any strong indication of feed by genotype interaction, especially when the feed is used as fixed effect in the model. So since the fish fed with different diets were full shifts between each other, we use the trait recording for each of the feed for the genetic evaluation of the whole number of fish. And then when the genetic evaluation utilizing data from one feed, the estimated value of the sheeps and their parents were estimated based on the performance of the sheeps in the other feed and vice versa. And we done the ranking according to that. And we observed that when you, for example, we evaluate the animals based on the control diet, both them and their sheep fed the different diet, there was very different effect, very different ranking. For example, the first animal in the control environment was 42 in the control diet, was 42 in the plant diet. And also, similar effect was on the growth between the April and July. So as we can see, for example, in the previous slide, there was a considerable ranking using EBV estimated from the growth results of sheep fed with one or the other feed. The ranking correlation was used in order to quantify this re-ranking, and there was considerable lower ranking correlation between EBVs of the same trait when different feeds were used. For example, in those 4 different traits, for the growth to April to July was the rank correlation is 0.73. From tagging to July, the growth period was 0.77. For the length was 0.74, and for the fat percentage was 0.76. So the rank correlations were varying between 0.73 and 0.77. So there is a significant ranking of the EBVs. And the genotype, the feed by environment interaction, is expected to affect 25% on average the selection decisions we're making. So we have to take into account this is the conclusion. But then we went to see what happened. For example, we use a genomic tool in our evaluation. What will happen, for example, we have genomic evaluation, genomic selection. From the next batch, we selected, of Cibrim, we selected 1,536 animals. We genotyped them with the Metafish array that is available. 671 animals, candidates, selection candidates, also with genotypes. And we used the Axiom suite for SNP polling and filtering, quality control. We performed a GWAS analysis only on the batch 17, and also we produce using BLAPF90 genomic estimated breeding values. From those animals, we present here the slaughter weight, which was on July and approximately 550 days post-hatching. There were 655 individuals from fed the plant diet and 773 the control diet. In total, 1,428 animals. The average weight at July was 445 grams in the plant diet and 501 in the control diet. We performed the genomic evaluation using a single-step GBLAB, using a BLAPF90 suite, and we estimate GBVs for the batch 17 and also for the selection candidate using the genomic relationship matrix. We have used the feed as a fixed effect as well as the different batch for the selection candidates and the batch 17. And again, the trait of interest was body weight in July. And we estimate the GBVs and also the direct values as they're coming out from the additive effects of the SNPs. What we notice is that on the above diagonal is the ranking correlations, below the diagonal are the Pearson correlations. What we notice is that this ranking correlation that we observed using the pedigree analysis was even lower in this case. For example, was 0.64 when you use all the animals and the feed is a fixed effect, when you use only the control feed fed animals, and the plant fed animals, it was even lower, 0.44. And when you compare, for example, the ranking using the ranking between the two different diets was a negative correlation. So that means that we have a significant different changes in the ranking using genomic evaluation. Genetic parameters of the trait studies in aquaculture are within the expected range that we saw from those experiments. from other published results and from previous estimates of the breeding program. The growth recorded in the same trait is the same trait at a different feeds, similar heritability estimates and very high genetic correlation. So we're not talking about genetically different trait, we're talking about the same trait. However, there seems to be significant genotype-environment interaction. We have significant family-by-feed interaction, considerably lagging with the individuals on the genetic evaluation procedure, and medium lagging correlations of EBVs. On average, 25% effect on the selection decision for the selection card is expected when fish are fed a plant-based diet. Measures should be taken in order to ameliorate such effect, and the measures we will see on the end. GWAS analysis performed showed possible fit by genotype interaction at the genomic level, and we're still working on that to pick up some SNPs that have significant interaction. And also, genomic evaluation is showing that fit by genotype interaction might be larger at the genomic level, so we have to be taken into account when we use this method for evaluation. Now, what's the solution? I think multi-industry has led the example. Okay, it might be costly to challenge, to evaluate with two different feeds, but we saw that the benefits from such a scheme in the improvement of the genetic gain is quite big. So I think it worth the extra cost for testing at the different feeds. I would like to thank, for example, funding from 3 different projects, from PerformFISH for a power project where the experiment with the 2 feeds were, another Horizon project featuring Aqua. And thank you very much to the colleagues from the HCMR and Mireos Aquaculture for their friendship and collaboration all these years. And also thank you for your attention, and thanks all my team that now is shrinking because we're losing funding. So many of them are in different groups across Europe, for example, some in Norway now, some here in Russia, I am in sabbatical. But thanks very much to all these young scientists that were brilliant. So then that's it for me.
Thank you, Dimitri. I guess we can reply to one question before getting to Valia for the last presentation. It might be both for you, Dimitri, and Mark. If selection for growth means that we will gain more protein.
Not necessarily, unless you monitor also the fat percentage. That's, for example, answering the previous question that was done in the first part. Always the breeding industry is recording more traits than those that they select. Even if you don't select for them, we need to record the traits. For example, in the poultry breeding, we know that with growth, you have to have, for example, strong skeletal structure. So we're actually measuring this before and afterwards. When birds were getting too big, we incorporate it in our selection. So we might lose some from growth, but overall we get more. And also, for example, when the change of the market was for more breast, we're reporting breast since the 70s, breast yield, breast fillet yield. We weren't selecting for it, but we're reporting it from 70s. So when the market changed, they demanded more breast fillet, the program was ready. So you have to anticipate these things, as Mark said, correctly said. And as far as the added genetic variation, some people, many biologists, they're worrying about it, that we lose the genetic variation. As far as the traits is concerned, we didn't observe something like that in the poultry breeding that I was working for a few years. Genetic gain was steadily growing, and we didn't lose any heritability after and we're talking about very heavy selection.
Thank you, Dimitri. Mark, do you want to add something? No.
I would say that for this question of gaining more protein, I mean, we are still in fish in the really beginning of selection for feed efficiency. So for the moment, we don't go to that really to that level of detail. For us, the real point now is to be able to measure feed intake, because if you don't have, I mean, protein gain, you can only measure it correctly in terms of efficiency if you know the feed intake, if you know the input of protein. So, it's not an easy question, but that's the aim. The aim when you do feed efficiency is to have more feeded, more protein to eat. Yeah.
One question that was asked by Greg Mice before the lunch break if we are aware of breeding programs that include epigenetics. I'm not aware of anyone in, at least for the Mediterranean species. I don't know for salmon or the rainbow trout.
No, I mean, at the moment we are trying to see how epigenetics makes things vary, and if this has some heritable component, but we are not at all at the point to integrate this into the breeding programs. No.
Yeah. And another one that has been asked for your presentation, Mark, is if there is a possibility to affect growth in the next generation when fish are selected for disease resistance, when the environment is the same.
In fact, it completely depends on the genetic correlation between disease resistance and growth. And for this, there is no general rule. I mean, you have some diseases which have positive correlation with growth, some have negative correlation with growth, some have zero correlation. And if you look at the big picture, there is absolutely no rule. I mean, so you cannot say generally selecting for growth will increase or decrease disease resistance. If I would say generally, there is no impact. But for some specific disease, there may be some impact in one way or in the other.
Okay, thank you, Mark. Valia, are you ready for the last?
We have some indication, for example, from challenge experiments in disease resistance in some, and also in parasite resistance, there is some positive negative genetic correlation, for example, but very small in some cases. Between growth, for example, those that are early fast growers, they have better disease resistance, especially in the early growth stages.
In bass, bream?
In, if I remember correctly, is bass or bream. I think it's either bass or bream. In Vibrio, I noticed that, or in VNN, but not hugely, not huge. There was some effect, but not.
As Mark mentioned, maybe there is no universal rule that.
I won't expect it, as Mark correctly said. I won't expect to be a universal rule.
Thank you, Dimitri. Valia, are you ready?
Yes. Hello. Well,
I try to share your screen.
okay. Can you see my screen properly?
No.
Okay.
I think we lost half of the presentation. I don't know.
Now it's better?
Yes, I think. We don't see the logo in full, but we may proceed.
Okay, just a moment.
So.
Is better now?
Let's continue. It's the bottom part, but I don't think that it will make much difference, Valia.
Okay.
We can go on, please.
Okay. My name is Valia Stavroula-Economou, and today I will guide you to selective strategies for disease resistance and parasitic diseases. I will emphasize on the European sea bass. And in the present agenda, we will introduce the disease resistance as a breeding goal, and 3 different breeding strategies will be presented. Moreover, examples for major QTL and genomic prediction in Salmonidae and in European sea bass for disease resistance will be briefly illustrated. Furthermore, 2 studies for the parasitic disease and in European sea bass will be presented, and finally a summary of the key points will also be shown. Over the last decade, infection and parasitic diseases have generated large economic losses in aquaculture farming. Thus, an increasing demand for the design of breeding programs to focus more on the disease resistance. So, apart from selecting fish with a higher body weight or better performance, fish with increased resistance to specific diseases should also be selected. This is a feasible solution to improve not only the productivity, but also the fish welfare. Other advantages of selecting the most appropriate fish for breeding for disease resistance over other methods against infection are the cumulative and the permanent benefit of the improved resistance. Disease resistance is used as a generic term to describe the ability of the host to limit infection or the consequences of the infection by reducing the pathogen replication. Other terms, such as tolerance, can be referred to the ability of the host to reduce the impact of the pathogens on performance without necessarily affecting the pathogen load. However, in this presentation, we will use the first term. So, fitting the disease resistance into a breeding program can be hindered by the difficulty to determine the measure accurate and appropriate phenotypes. Generally, phenotypes can be classified into 2 main types of data, categorical and numerical. In the first group, the nominal is the most popular. In this case, we identify if the fish is dead or alive. Other types of data can be classified into numerical, which also includes the discrete observation. Those types of data are not normally distributed, and we need to transform them into a logarithm in order to use them. The way we collect the phenotypes are obviously to challenge the fish with the disease, which is costly, time-consuming, and requires a trained staff to do it. When a disease challenge is performed, the population is divided into the reference population and on the selection candidates. In classical breeding programs, the reference populations and the selection candidates are seeds, for example, full seeds. So the reference population is used for the disease resistance, and the outcome of the disease is measured on them. Then the groups of the reference population and the selection candidates are united, and the phenotypic records of the reference population are used to train the model, and the breeding values are estimated for the population. However, only candidates from the selection candidates groups are used as a breeder. Let's move on the model that we use in order to perform selective breeding, which can be done using the classical approaches.
phenotypes from the reference population are recorded, and they are combined with the pedigree information using the BLAP, which is an equation, and the solution of the model are the breeding values from the reference population, while the prediction of the breeding values for the selection candidates are also estimated based on the information of the reference population. So the reference population is used to train the model, and the breeding values of the selection candidates are calculated based on them. Then we select the fish with the better breeding values from the group of the selection candidates. Until now, breeding programs have included disease resistance based only on information from reference population, which is related to the selection candidates. This affects the degree of the genetic process achievable in each generation, and as a result, lower accuracy of estimated breeding values since only seed selection is available. Other disadvantage is that if the trait shows low inheritance or if it is affected by multiple genes, then this method is less effective. Some of those disadvantages can be overcome using marker-assisted selection, which is used for the indirect selection of the fish, which are superior. In order to perform MAS, it is mandatory to identify an association between a genetic marker, which is linked to a quantitative trait locus, and the outcome of the disease. This genetic marker can't be used in a selection program. So, in practice, fish were genotyped using microsatellites or SNP markers, and the phenotypic records were collected and used in order to perform GWAS or QTL analysis. The results are plotted, and if the PEV value of the highest point is lower than the selected threshold, it is considered as a statistically significant marker. So our next step is to identify the effect of this QTL on the outcome of the disease. Is it a major or a minor QTL? Thus, we estimate the genetic or the phenotypic variance, which is explained by it. To sum up, MAS involves scoring indirectly for the presence or absence of the desired phenotype based on the sequences of the markers located in or near the genes which are controlling the disease. Thus, the sequence of the marker is indicative of the presence or absence of a specific gene or a chromosomal segment which is not to carry a desirable allele. If a major QTL is available, the advantage of MAS is that there's no need to perform disease challenge along with the higher genetic gain. The selection accuracy and the heritability of the trait are not under consideration. However, if the outcome of the disease is affected by multiple genes, and each of them has a small effect on the phenotypes, or there is not known QTL, then other methods, such as genomic selection, can be used. Genomic selection can improve the genetic gain and the selection accuracy. This alternative technology uses all the available genomic information from the SNPs, and it provides a major advantage, since all the QTLs are likely to be in a linkage equilibrium with at least one SNP. In this case, the genomic panel and the phenotypic records are required. From the genomic panel, the sum of the effect of those SNPs across the entire genome is calculated based on the phenotypes, and the genomic breeding values are estimated. Generally, genomic information provides higher accuracy of genomic breeding values, and it can be performed early in fish life. So the benefits of the genomic selection include a more accurate estimation of the genetic relationship between any two individuals that could be given by the pedigree records alone. However, the cost increases due to the high density genotyping in a large scale of individuals. In practice, an increased accuracy of genomic prediction compared to the pedigree, up to 22% for disease-resistant trait, had been noted. This means that the use of the genomic selection provides better, more accurate prediction compared to the classical approaches. To sum up, the genomic selection is superior to the classical approaches because it provides more accurate estimation. Thus, it increases the genetic gain, it controls the inbreeding better, and it decreases the generation interval. However, it is expensive to apply because of the genotyping cost and the purchase of the SNP arrays. Moving on, the mass is superior to the classical approaches because it provides more accurate estimations and increases the genetic gain. Also, it reduces the production costs since no challenge test had to be done, while genotyping for selection candidates can be performed in a limited scale. However, a major QTL has to be found in order to be able to follow such a selection strategy. Finally, if a major QTL is known, mass could be superior to genomic selection, since no challenge test is required to be done and genotyping cost can be achieved in a limited scale. However, if the trait is affected by multiple genes or no major QTL is available, then genomic selection is superior to the mass. All those strategies have already been used in aquaculture. Disease resistant as a breeding goal has appeared for over 20 years, and the first breeding program which has focused on it was in salmon in 1993. A successful example of mass is the case of the infection pancreatic necrosis resistant in Atlantic salmon, in which a major QTL explains the majority of the genetic variants for resistance, and it has been demonstrated as a successful means of controlling the disease. Since selecting fish with the preferable genotype in this QTL practically halts fish mortality. Apart from mass, a comparison between genomic accuracy using genomic information and using pedigree information has been done for disease resistant in Atlantic salmon and in rainbow trout. Some of those studies are illustrated in the table. The findings show a higher accuracy of prediction using genomic than using pedigree relationship matrix. To sum up, all the genomic information provides better estimates for disease resistance, and higher genetic gain can be achieved using it as a breeding goal strategy over pedigree. Apart from Salmonidea, which also show the highest production, Mediterranean species have also have a significant impact on fish consumption in Europe. One of those is the European sea bass, in which several types of diseases have been studied, but in a limited scale compared to Salmonidea. Focusing on those studies, only a major QTL has been found against VNN, while other diseases, such as vibriosis and parasitic infection, have been studied. A trial to interpret the benefit of the genomic selection over the pedigree approach for VNN and vibriosis has been done. showing the superiority of the classical approaches. Focusing on the major QTL against the VNN, several studies have been done. And the last one shows that approximately 33% of the genetic variants of the disease resistance can be explained by this QTL. This final evidence revealed almost a perfect mass scenario for the disease resistant against VNN in the European seabass. Apart from VNN, other diseases which are popular in Greece and Turkey are the parasitic ones. Parasitic diseases may not cause direct mortality, but could increase the production cost through treatments or reduce the quality of the product, and may also have a significant impact on growth and fish behavior. A popular ectoparasite is the Lenanthropus cruielli, which has affected the European sea bass in Greece and Turkey. The main problem is that the fish cannot breathe properly if there are a large number of Lenanthropus cruielli attached on the gills. And generally, mass mortalities due to it have also been reported due to heavy parasitic infestation. Apart from Lenanthropus cruielli, another parasite which also attaches itself on the gills of the fish is the Dipelectanum equans. The main problem which is created by the infection of Dipelectanum is that it inflicts a significant injury to the gills filaments, which can lead to a secondary infection from other pathogens, such as bacteria, viruses. However, the heritability and the genetic parameters between resistance and growth are not known. To study the Lenanthropus cruielli, 2,425 European sea bass were put at Sayada site. And after 6 months, in the sea cages, all the fish were sacrificed. Fish infection was conducted by natural cohabitation with the parasites at the designated sea cages. In simple terms, fish infection was natural in the Sayada site. No artificial infection was caused by human. Then, expert members of Nireus pathology department counted the parasites on the gills of the fish using stereoscopes. So for each fish, the total number of parasites was available. And their average number was 24. Focusing on the heritability of the parasite count was 0.28. The parasite has a low to medium unfavorable genetic correlation with the body weight and growth. But it seems that it is not significantly impairing selection for growth. Moving on, to study the Dipelectanum equans, 4,450 European sea bass were put at Nafpachtos site, and after 6 months in the sea cages, all the fish also were sacrificed. The trials were conducted twice in 2 consecutive years, and also fish infection was induced by natural cohabitation with the parasite at the designated sea cages. And the parasite were also counted using stereoscopes. Their average number was 26 and 47 for the first and the second year, respectively. Heritability of the parasite was medium equal to 0.2, and the parasite does not seem to affect the body weight, focusing on the phenotypic records. But the genetic correlation between the body weight and growth seemed to be slightly related with a high parasites count. All those results were published in the Journal of Aquaculture report, and based on the results of the Lenanthropus cruielli, which was very promising in terms of the existing potential for genomic improvement for parasite resistance, and provides a good base for further genetic analysis using genomic selection, we perform another experiment, and we use the same fish infested with Lanthanthropus cruielli in this analysis, but we performed the selective genotyping in order to reduce the cost of genotyping. To do that, we used all the phenotypic data and pedigree information, and we performed lab, and the EBVs were estimated, and a specific number of fish was selected, using firstly the within family variants of the parasite count, and secondly the discordant EBVs for the parasite count. The selective genotyping was applied in such a scheme in order to include both within and between family variation in our sample. And the selected sample was genotyped using the 30K matrix Metapisys MP array. The genomic data will be filtered using the quality criteria which are illustrated in the board. And after the quality control, we performed a GWAS in order to identify SNPs related with the resistance to Lanthanthropus cruielli. We identified two putative SNPs related to it, but they were near to the threshold line. For those SNPs, we calculated the descriptive statistics, and approximately 4% of the phenotypic variation can be explained by them. But this percentage of variation, which is explained by those QTLs, can classify them to minor QTL if we remember the Manhattan plot from the resistance against BNN. Thus, for Lanthanthropus cruielli, the mass is not a preferable option due to the low effects of the detected QTLs. Further studies are underway in order to detect the genomic area near them, which could be considered as a major QTL. However, a readily available option could be genomic selection, which will be presented in the next slides. For the classical approach, the pedigree relationship matrix was used, and the phenotypic records were combined in order to estimate the breeding values only for the selective sample. On the other hand, the genomic information was used in order to estimate the genomic breeding values. The population was divided randomly into training set and validation set. In the validation set, the phenotypic records were masked, and the predicted breeding values were estimated using the pedigree or genomic relationship matrix based on the method. This process was repeated for 20 times, and each time a different validation set was selected. So only for the validation set, the correlation between the predicted breeding value and the phenotype was estimated for each method. Then a pairwise t-test between the two methods was calculated. Moreover, the accuracy of the prediction using only the validation set was calculated using the correlation between the predicted breeding value and the phenotype divided by the square root of the genomic heritability. The first table illustrates the descriptive statistics of the correlation between the predicted breeding value and the phenotype only for the validation set. And the plot illustrates the same information graphically. The average correlation between the genomic breeding value and the phenotype was greater than the average correlation using the pedigree information. In this plot, the lines between the repetition were drawn, and a pairwise Sample t-test showed a statistically significant difference between the two groups. Thus, using the genomic information provides us a better estimation of the breeding values when the phenotypes of the fish are missing. Those findings show us that using the genomic selection, a higher accuracy can be achieved compared to the pedigree selection. The increase was equal to 17% between the two approaches. To sum up, the key points of the experiment of Lenanthropus croyeli revealed two putative QTL related to resistance to Lenanthropus croyeli, classified them as minor QTL. Thus, mass selection will not provide a great improvement. An alternative breeding strategy is genomic selection, since a statistically significant difference between the predicted EBVs and the predicted GBVs is estimated, along with a higher genomic prediction estimated equal to 70%. So, if the resistance to Lenanthropus croyeli is used as a breeding gold in Mediterranean countries, the use of genomic selection could provide higher genetic gain, because higher estimation can be achieved for the selection candidates based on the phenotypic records of the reference population. Resistance to diseases can be defined as the ability of the host to limit the infection by reducing pathogen replication. Disease resistance can be used as a breeding gold in commercial breeding programs. In some cases, such as in Salmonid, it has been used since 1993, while in others, such as European sea bass, it is limited until now. In European sea bass, a major QTL has been found to affect the resistance against VNN, so mass selection will be applied in the future. And the method to select the fish with the higher resistance against pathogen can be performed using pedigree selection, which provides small profit, can be performed using mass if a major QTL has been found. And this method will be the most cost efficient, since there is no need to challenge the fish. And finally, genomic selection provides better estimation of the breeding values if no major QTL have been found, and if the trait is affected by multiple genes. All the experiment and the analysis of Lenanthropus croyeli and Diplectanums equans have been funded by the project PerformFISH. And thank you for your time.
Thank you very much, Valia. It was an excellent presentation.
Thank you.
General for the topic, but it is true when we will try to squeeze everything for disease resistance, it cannot be done in half an hour, 40 minutes presentation. I think we could wait for a couple of minutes to receive any question in the Q&A window. I would also like to thank all the participants, you, Valia, Sarah, thank you very much, and Mark twice. I'm going to thank Mark twice, each for one presentation, and Dimitri for putting the so much time and effort to facilitate the PerformFISH Work Package 1 webinar. I don't know, Mark, do you wish to say something? Dimitri, Valia?
Well, I just want to say I'm very proud of Valia. It was really nice for the presentation. I really enjoyed it.
Thank you.
It was, I think Valia's presentation was better than yours, Dimitri.
I totally agree.
Yeah, it was really, really good. And I think as a team, we've been quite good because we have not seen each other's presentation before today. Yeah? We had discussed on the titles, and just with the titles, we managed to have excellent complementarity. So I think it's really nice.
The overlap was very minimal. I mean,
yeah.
It was a brilliant flow, I think. Brilliant flow, very nice.
Okay, I don't know. I think that if anyone had the question, he should have posted it by now. I'm not aware if Keegan or the Aquaculture team would like to intervene and tell something or guide us to what to do next. I would also like to thank the participants, the attendees that have been present into this course. For the time being, 52 have had the courage to stay for so many hours. I think that the original numbers were quite high, approximately 85 or 90, if I remember well, at some point. So we should all be proud as panelists, I mean, as people that prepare these presentations. And I would like to thank you all for attending the meeting, and of course the people that prepared all the presentations, and especially the younger scientists. And it's not certainly you, Dimitri.
I don't think so. Cells are some things from almost a quarter of a century ago.
So
when I said, when was that? I said, wow, it's been so many years. Yes.
Sincerely, thank you, Sarah and Valia, and again, Dimitri and Mark. So looking forward to see you. Unfortunately, not in an annual meeting of PerformFISH. This will be probably done physically in Rimini in the next EAS meeting. So looking forward to see you there. And I'm also aware that few of us will be in the Genomics in Aquaculture symposium in Granada next month already. I know Valia will be there. I don't know for you, Mark or Sarah. So unfortunately,
no, I won't be in Granada actually. I will be in Rotterdam.
Okay, so
at the World Congress.
Yes, you're right, because it's also a very big meeting for animal breeding in Rotterdam this year. Yeah.
Yes.
So thank you all. Nice to see your faces, at least the 4 or 5 of us, and it's unfortunate that we couldn't open it up to everybody, but it's not easy to handle. At least I cannot easily handle it with so many participants. It's a pity not to see 90 more faces, but okay, that's life. Thank you all for attending.
Thank you, Kostas, for the organization. Yeah.
Thank you.
It wouldn't have happened without you, Mark, and Sarah, and Valia, and Dimitri. Yeah? Thank you all.
Thank you very much.